everyone. Now I think we have resolved the problems with the sound. And now it's time to continue our symposia on Coralligenous with the third session that is about impacts and disturbances of this K habitat. The first presentation is Kim Garabu, as you see, and this is the T-MedNet Climate Change Coastal Observation Network, tracking and assessing effects on thermal regime and their effects in Mediterranean coastal ecosystem. Kim, floor is yours.
Thank you, Besna. Good morning, everyone. I'm really happy to see that after the great gala dinner, many people are here. I thought that we were a small group, so thank you very much for getting up earlier and being here. Oops. So I take this time to thank everybody from the organization and my colleagues from the scientific committee that have been chairing so well the different sessions. Okay, let's go. So I'm going to start by the origin, because sometimes we discuss about things and people don't know why we were doing this. And this is, I want to give you some information on why we started to work on this network, the T-MedNet network. And in fact, more than 20 years ago, we were witnessing for the first time a large-scale mass mortality event in the coast of France and Italy. And this event affected many species, around 30 species from different groups. And what we realized is that the main cause, after some analysis, is probably it was linked to the high temperatures that had happened in that year, in that summer, in '99. But when we were looking for the temperature data series, this basic parameter, temperature, I mean, measuring temperature, we realized that there were just a few temperature data series around the coast. And in fact, the only high resolution temperature series that they were initiated by Jean-Georges Armelin. And here I'm going to stop and making a parenthesis, just saying that maybe as a community, we should think about paying some tribute to these pioneers like Jean-Georges Armelin and Carlo Nicchio Bianchi. As a community, maybe we should think about this. And even the people that sadly is gone. And as a community, I think that we need, to remember our members and to see, make tribute to these people. So these pioneers, among them, Jean-Georges Armelin, started to set up this temperature data loggers in Marseille, but it was only one point, at least known in the Mediterranean. And 20 years ago, and this figure shows you the coverage of the marine heat waves in the Mediterranean, and you see he pointed the year '99, and the different colors, it's the category of the marine heat waves, so from the strong to severe to extreme. But 20 years ago, we saw this increase, but now with the next years, still even this year, the marine heat waves impacts are so great and has been increasing and increasing and increasing. So we have an increase on the frequency of the marine heat wave, on the extension of the marine heat wave, and the intensity of the marine heat waves. So the climate change impacts now are overwhelming and are, we cannot understand what is going on in the Mediterranean habitats without taking into account the climate change dimension, right? So this is a call as well to everybody here and everybody in our actions, in our activities to take action somehow, try to contribute to stop the climatic emergency in which we are living, unfortunately. So it's in this context where we started to work on the T-MedNet network. At the beginning, we didn't have a name, but we started to work in this network, and we have this. You can read the vision and the mission, but the bottom line of this is to build a collaborative network of people, organizations, managers, researchers working in the coastal areas to track what was going on with the climate change in the Mediterranean. So the philosophy behind all the protocols or the activities that we were working is that they had to be really practical and cost effective, try to be standardized or at least harmonized. And it has to be something that we can apply at the regional scale. So at the regional, it should be a regional strategy to have this global view on the Mediterranean. And it had to engage people, the local people, because we cannot think in a single team going everywhere in the Mediterranean taking measures. We have to engage local people, and in this way we will be much more effective and efficient in the implementation. So I consider, or the people involved in the T-MedNet, we consider that this is a successful collaborative story, probably not so great as well we wanted, but we consider it a successful collaborative story. And here I put some indicators of success, of how we can measure if it's successful or not. So along the next slides, you will see some numbers about the number of people, the organizations, publication, observations, and this you can have your own criteria if this is successful or not. So what I'm going to present today here is the results of the implementation of 3 monitoring protocols. The first one, it was related with the origin of the T-MedNet, which is the measuring the temperature. So we set up this temperature data loggers at the different depths, at fixed depth, so every 5 meters, from 5 to 40 or more meters, and then we set the data loggers to measure the temperature every hour, and in this way we have this really nice high resolution temperature data series. And one of the criteria for me, at least success, is the feedback from the people that is using these tools. And I take this message that it has been sent to us by an MPA manager in Briuni, in Croatia, that explains that she's using the tools that we developed, and for her it was easy and really helpful to have this. In this case, it was the video tutorials explaining step by step how to implement the monitoring protocol. And if we look at the overall results of the implementation of this monitoring protocol, we have you see in these figures on the temperature series that we have in some cases more than 20 years of data, recording the data in different sites. We can compare the different temperature regimes in the different areas, so it's really useful information. There are more than 20 million data points of observation of temperature, and if we put this in one single temperature series, it could be like having more than 2,000 years of measures. So I think that it's kind of impressive work. This has been implemented by more than, I put here big numbers, but more than 100 participants among MPA managers and researchers that have been implementing this protocol in 11 countries around the Mediterranean. The second monitoring protocol that we have been implementing is the monitoring mass mortality protocol. And this is linked to the presentation that yesterday Laura Figueroa gave you about the rapid assessment. So this is the idea of implementing this protocol. So just to get an information get information about the impacts of the mass mortality. So we publish a collaborative database paper, and we gather for the first time more than almost 800 records of mass mortality events in the Mediterranean. We have a web platform that I invite you to explore, and where you can see the data, you can download the data, and if you have information that it's not in the database, you can upload this data as well. So join the network. And one of the major outcomes of this approach on the mass mortality is this year we have published this paper in Global Change Biology about the mass mortality events occurring in the Mediterranean between 2015 and 2019. And this has been a huge effort by more than 35 research teams from 10 countries. We got more than almost 1,000 monitoring surveys on mass mortality, and we have this really nice image across the Mediterranean. Of course, there are still gaps, and this is something that we have to fill up. And linking the information of the temperature and the mass mortality, it has been really useful to have this temperature series. And here I put two graphs from the same paper that I invite you to visit as well, about the warming rates and about the marine heat wave days across depth, because the previous information, most of the information about marine heat waves is about the satellite information, satellite data, and we know that the impacts can go down in depth. The third and the last monitoring protocol that has been implemented in T-MedNet is the climate fish, which is coordinated by Ernesto Azzurro, who is here in the room. And it's about tracking changes in fish species that respond to the climate change, so the changes in temperature and marine heat waves. So there is a selection of target species for this, and then we with this it makes some transects, standardized transects, and in this way we can see the transition from towards this tropicalization or towards a more thermophilic fish community in the Mediterranean. And there has been as well a publication on the data and on the method of this climate fish. And here you have the number of transects that have been carried out in different areas of the Mediterranean. So more than 3,000 transects from different ecoregions in the Mediterranean. And in some cases there are as well temporal series acquired, and in this way we can track the changes. So I'm just heading the end of the talk. I hope that I'm not too long. What are the next steps? The first thing that I want to do is to make a new call to quantify the 2022 mass mortality impacts, because we have been witnessing, and I had the opportunity to exchange with some of you during these days about these impacts. This is an image that I had the opportunity to go with a team in Marseille, Septentrion Environment, that organized these field actions with the Parc National of Calanques. And this it's overwhelming what is happening there in Marseille, but in other areas of the Mediterranean, it's something maybe not so dramatic, but it's something that is happening. So this is a call to collaborate here in knowing what is going on with this. The second next step, that is a long-term issue that we have been working, is to expand the network and establish a sustainable functioning of the network, because we need support. Like yesterday we were talking about the citizen science initiative. We need some funding, we need some support to keep going with this activity. So we need to fill all these gaps across the Mediterranean and get this image, a global image about what is going on with climate change in the Mediterranean. Of course, we need as well, I talk about 3 dimensions of the climate change: temperature, mass mortalities, and shift of in the distribution of fish species. But there are others. There are changes in the reproduction. There are problems with Posidonia. There are other species that are shifting distribution. So we need to expand as well these protocols. So this is as well a call. I'm making a lot of calls here to join and support the network, because I think that it proves that it works, but we need to keep going and expand and make it happen. And this is the last next step, and it's a proposal that we are trying to put together now. That it's It comes from these processes. I don't know if you are familiar with the roadmap of the MPAs in the Mediterranean. And as well, I had the opportunity to be the coordinator of an InterRegMed project. It was called MPA Engage. And we had a final event and we make a list of recommendations. And among those recommendations, it was to take the MPAs as sentinel sites for climate change. And in a way, I thought that maybe we need something concrete and ambitious at the same time. And it's something that is still to be discussed, but it's maybe we can launch this challenge about having 100 MPAs we have in the Mediterranean more than 1,300 MPAs. So only 100, but if we have already 100 MPAs by 2030 that are conducting some activities on climate change, I think that it would be a really nice challenge too. And for this, we could start working with the SPAMIs, so these MPAs of special protection, and from this in the different countries and start building or reinforcing this activity of. So I just want to thank everybody that has contributed to the T-MedNet network in any way. And I forgot to mention that in the authors we were only 3 names and then the T-MedNet network, but I know because the list of people, it couldn't fit in the rules of the proceedings. But I want to thank you. So a round of applause for everybody who has been participating in this. And thank you very much.
Thank you, Kim. We are on time and we have time for a few questions if you want.
I forgot to mention just that we bring, this is one of the products that we did in T-MedNet, which is the description of the several monitoring protocols that with Ernesto Azzurro, I don't know, we can leave it somewhere for the coffee break. And we did as well this Ernesto has done this really nice book, illustrated book about the climate change and the role of marine protected areas in the Mediterranean, which is called Boiling Mediterranean. I think that is really timely. So there are some copies as well that if you want to take one. Do you want one?
Yes. Okay. Well, I could take a floor and say that as we have a new MPAs in Montenegro, so I promise you will have data from the South Adriatic in the next year. And I think it's really important to have a really huge network because the data are not so optimistic, I would say, but we should do what we can do. Mm-hmm. Any question?
Okay.
You are clear. Okay, so we have a question. Please, Mehdi.
Thank you, Kim. It's not a question, actually. It's a comment about the last call you made about SPAMIs and MPAs to be first sentinel for climate change monitoring. We can promise you to support and to coordinate that.
Okay, thank you very much.
Perfect. This is good news.
I take the opportunity that I have the mic here close to me. Monica Montefalcone, University of Genoa. I just would like to share with you our recent experience. Kim knows very well that we developed 2 years ago a net of Italian marine protected areas in a project called Mare Caldo, Hot Sea, which today see the participation of 12 marine protected areas in Italy, which are going to follow the protocol of T-MedNet. We developed a system of monitoring of water temperatures in all these stations. So I think that for the Italian nation is an important achievement. Of course, we can develop it a little bit more, but what I would like to to say to all of you, and especially to you, that you say that you are going to support us, is that we need some support because marine protected areas are very happy to join this network. The network is working a lot, but probably if we have a support by SPAMI and something like this, just to help us to keep on growing the net. Each marine protected area is able to support itself because, you know, this kind of monitoring in terms of thermal sensor is not so expensive, absolutely. So each area was able to provide the financial support to come inside this network, but it's important for us now to be able to go on with this project. And if we have some, just a support, not economic, I mean, just to know that we are known by everybody and we can keep on going like this. So
don't be shy and ask for financial support.
Yeah,
of course. Finally, it could be nice also.
And may I add also talking about support, it is not that only you will leave the thermometer somewhere, you have to go to clean it. I unfortunately lost 2 winter times because of epiphytes on it, but so it is not just stay there 1 year and then you will have data. So we need support in every case, yeah. Okay, thank you, Kim.
Do you
Maybe
want
we
this
can...
one?
You have a question, Sylvia.
Yeah, no, I don't have a question, Sylvia Gibson. But I want to make just a comment. I mean, of course, MPAs are essential partners in this, but we should not also neglect the other potential ones. I can give you an example of a dive center in Croatia who completely self-financed temperature data loggers, the small ones, and they put it in their sites where they're leading touristic tours almost every day. And it's part, I don't know, I would say citizen science or whatever, but they were really willing to do it. And for them, it's really interesting because they can also show this to their clients, to divers. So they're actually spreading the word as well. So I think they could be also really important partner in this initiative.
Yeah, I forgot to mention that the idea, as yesterday in Cristina Linares' presentation, the validated data from citizen science, it has to be as well, the idea is to incorporate this, the validated data in the databases here in T-MedNet. So it's gathering all the information. And of course, we are focusing on MPA managers and scientists because are the kind of the target community. But there are here in Portofino, there's a group of diving centers that they created an association in this sense. Right? I think that it's called Coral Alert or something like this. And I think that it's an initiative that it came from the diving centers, and they are taking the reef check protocols, the Observadores del Mar protocols, and they do trainings and things. So I think that it's really an important way of increasing, contributing, because I mean the climate change emergency asks for everybody's effort and everybody's expertise. It's not only one shot thing. I mean, we need the different the efforts from everybody, and we have to find, everyone has to find the way on how we can best contribute to the transformation of our society. And in our case, I think that at least in my case, the T-MedNet network is the contribution that I'm working on this, and Observadores del Mar, it's a contribution that I'm making on this, just to make gathering information on what's going on, because if we don't know what's going on, it's really difficult to know what to do. And the second thing is to raise awareness on the people, because even only this, it's so important. Now the people, they felt the 2022 summer heat waves and everything, and they know that something is happening, but still, I'm just take one more minute from your time, but I was listening about the skyrocketing prices of the fuel with the high temperatures in July in Spain, and the news where there were more than 50 kilometers of traffic jams in the highways. So, I mean, we have a problem here and we have to try to solve it. Thank you very much. I stop.
Thank you, Kim. So we will continue with Yanis Zentner, climate adaptive management in marine protected areas. The case of marine heat waves and diving frequentation on the conservation of Coralligenous habitat forming species.
Okay, give it to me. It will be the only poster, so it should be easy to find. Okay, thanks a lot. Good morning. My name is Yanis Zentner. I'm a PhD student at the Universitat de Barcelona. And today I will be presenting one of our latest study where we looked at a possible climate adaptive management in a marine protected area. In this case, as said, focusing on marine heat waves and diving frequentation and the negative effects that they were having in one of its key habitat forming species, which will be Paramuricea clavata. So in the current context of climate change, the big question is what will be or what is the role of MPAs? And while there is a very big concern that warming will surpass any enhanced resilience they might be providing, they have also been proposed as one of the tools we have to try to delay and mitigate the effects of climate change. And what is clear from both perspectives is that there is a general lack of quantitative assessments that truly calculate the benefits of having them in front of climate change. So whenever we try to do such an assessment, we need to acknowledge that many are still locally threatened. Those that are not strongly protected due to a lack of enforcement or due to authorized destructive activities. But even in strongly protected no-take marine reserves, we have some recreational activities such as scuba diving that, if uncontrolled, can have an impact on their communities. And another added difficulty to trying to do such assessments is that many times, if you have multiple stressors at the same time, it's difficult to distinguish which negative effects belong to each of them. So we need monitoring that allows us to individualize these impacts. So in this context, we find ourselves in the Medes Islands. It's a small no-take marine reserve that is very crowded by divers. Already in the past, it's been quite detailed described that diving is having an impact on Paramuricea clavata by an involuntary detachment of their colonies. While previously to the study, we didn't find any records of a mass mortality event occurring due to marine heat waves. So in the last 6 years, we perform a monitoring through 6 permanent plots where we individually tracked almost 700 colonies. And from them, we measured the maximum height, and we also evaluated their surface, the injured surface they had, distinguishing between recent or necrosis injury, whether the new taxis is still visible, and then when it is an old injury and this new surface is already overgrown by epibiotic organisms. And from this, we can calculate a set of demographics such as density, biomass, or annual mortality rates. And we also use the individual tracking of the colonies to project their long-term viability through size structure matrix models. And we use the life cycle previously described for these species by Linares et al. In 2007. And by following the individual fate of all colonies, we can then compute their survival, growth, shrinkage, and fecundity rates, which then get translated into annual transitional matrices from which we can calculate growth rate, do long-term projections, and even simulate some scenarios. And this has been used in the past by focusing on 2 different populations, one affected by marine heat waves, the other one by diving, to simulate the interaction that these 2 impacts have. And as you will, a small spoiler, you will see in our results that they have a very strong synergic effect. So what has occurred these past years? Well, I think we can all agree they have been quite eventful. And for the Medes Islands specifically, in 2017, we had the strong mucilage outbreak that entangled many Paramuricea colonies.
we have been seeing a slow increase of marine heat waves. Here you can see, thanks to the T-MedNet initiative that Kim just explained, the in situ data at 15 minutes and the marine heat waves and their intensity throughout the years. And we can see that the recent years, 2017, 2018, 2019, start to have more and more marine heat waves. And on the other hand, thanks to COVID, we had a decrease of divers, but we were very unlucky that on the same year, we had one of the strongest storms, historical storms that has occurred on the Western Basin. So we haven't been able to detect any benefit of this diminishing of divers. And when we look at the demographics of Paramurithea for these 6 years, there has been a very strong reduction, both in the density and in the biomass, while the mean height has maintained relatively stable. And if we look at the injury data, we can see a clear peak in 2018. This peak would fall in what we now classify as a moderate mass mortality event, looking at the amount of colonies that were affected. And this could explain some of the density in biomass we've seen, especially for that transition, 2017-2018. But in general, if we look at the injury data and the density and biomass loss, there is a small mismatch. Of course, this can be enlarged by these two other extreme climate events we've mentioned. But this is a first sign that there is another stressor at play, and this would be the overfrequentation of diving. Here we have annual mortality rates for these populations, and we have distinguished between two types of full mortality, which would be an overgrowth when the colonies are fully injured, and then a detachment when the colonies disappeared from one year to the other one, but they were still alive on the previous year, of course. And we are quite lucky because we have data from this exact MPA before the 2000s, before any presence of marine heat waves. And if we compare them, looking first at the overgrowth, of course, we've seen an increase of overgrowth, which we can relate to this increase of marine heat waves. But what is quite interesting is that if we look at the detachment rates, especially considering that this diving quotas have maintained exactly the same for these two time periods, we can see an increase of detachment only by the presence of marine heat waves. So it seems that its marine heat waves are facilitating the detachment of the colonies involuntarily done by the divers. This is especially clear if we look at the detachment rate of colonies, depending on how injured they were the previous year. The more injured a colony is, the easier they get detached. This again gets reinforced if we look at the population growth thanks to the matrix models, where we again have data before any marine heat waves, as the first one detected from the temperature analysis was in 2006. So we would still be before that. And we can see, of course, that the presence of the two impacts has worsened the population growth rates. And when we randomly draw the different transitional matrices and project them long term, we can obtain a stochastic growth rate, which again is lower. And if we visualize these projections, we can really see the catastrophic effects that these two impacts combined have. First, if we look at the probability of quasi-extinction, which would be the 10% of the initial population, with both impacts, it's projected that these shallow colonies, which are around 20 meters, 15, 20 meters depth, will disappear in the next 20 years, which is almost 30 years quicker than when we only had the diving impact. Something that is quite useful from this matrix models is that we can try to simulate some different scenarios. And what we try to do is differentiate the two impacts. For the global stressor, what we did is change the probability of having an extreme climate event. Of course, for the data that we acquired for these 5 years, we had 3 in 5 years, so it's quite unlucky, but with a future projection look, it's also not too far from reality, but we can change this probability to what we think will be the expected. And then the local stressor, we basically change the detachment rate that was caused by the diving impact. And when we project the long-term viability for all the possible combination of these two stressors, we obtain this graph where we first again see this strong synergistic interaction. But this also gives us an opportunity to try to say, okay, we want to go back to before marine heat waves occur. So what do we need to do? What can we do locally? So if we were able to reduce detachment in around 50%, we would be at those growth rates previous from marine heat waves. And this is not only through reduction of diving, there's other strategies that can be implemented as well to try to lessen the impact of divers. But there's also, in this very strong synergic interaction, there's also opportunity to be seen, because the worse the climate situation becomes, the more beneficial it will be to actually be able to obtain a reduction of the detachment. So there is, of course, the long-term projections are not very optimistic, but it reinforces that there is something we can do. There is something in our hands that we can do to try to fight against it. So in general, here we want to first really put an emphasis of the strong interaction that there's between this local and global impact, and that is having a very important decline for Paramylithea clavata within this MPA. But we can also say that through an adaptive management, climate adaptive management, we are able to delay the effects of climate change. And it's really important to have this type of quantitative assessments that use biological criteria when we want to try to design climate responsive strategies. So thanks everyone, a special thanks to my supervisors for all their support, and thanks to everyone involved in tracking these 700 colonies throughout the years. I will be happy to respond to any questions or comments.
Thank you, Yanis. Perfectly on time. Do we have questions? I think it's example how we really can do something. Yeah. Although we speak that climate change is not up to us small people, but I think it's really good example. Yeah. Questions? No. Okay.
Thank you.
Thank you. Thank you, Yanis. So now, hopefully, we have online Ilaria and Ilaria. Could you hear us?
Yes. Good morning.
Perfect. So, could you share your screen?
Yes. Okay.
Almost. Perfect. Now.
Is it all okay?
Yeah, full screen. Yes, yes, just full screen. Put.
Okay.
Okay. No, it's still on, not on the presentation mode. Full screen, please.
See, full screen.
Yeah, down. You were down on the right, the full screen icon.
Okay. I see the full screen.
Well, here is not.
It's F5.
I don't know. This is full screen.
Do you use double screens on your computer?
On my computer, I see the full screen.
So please, could you share that? That one, yeah.
No. It's okay now?
No, it's the same like previous.
I see the full screen on my computer.
Well, maybe also we can continue like this.
Ilaria, I can share your screen from me, and then
Ilaria, scusa, stai usando due monitor per caso?
No, ho solo un monitor e io ho fatto la condivisione dello schermo e ho tutta la presentazione con full screen. Io lo vedo così dal mio computer. Cosa devo fare?
Allora, togli la condivisione, metti la full screen e poi condividi.
Allora, io ho messo, ok, un attimo. Come prima ho fatto. Quindi condividi schermo, seleziono la mia presentazione, metto full screen. Lo vedete?
No, vediamo ancora l'anteprima, cioè a sinistra vediamo le slide e a destra la slide principale.
Quindi rifaccio, interrompo la condivisione. Poi
Metti prima in full screen e poi condividi la pagina in full screen.
scusate, rifaccio. Aspetta, Oddio.
Ilaria, se è un problema condividiamo noi la tua presentazione da qui.
Sì, grazie, perché io lo sto facendo in...
Ok, perfetto.
Could you see it now? Ilaria?
Hey, okay, I'm here.
Could you see your presentation?
Yes.
Okay, so please go on.
Okay, thank you. Sorry for this problem.
Don't worry.
Good morning, everyone. Thank you for giving me the opportunity to present our work. My name is Ilaria Medeo. I'm a biologist, and these are my collaborators, Canessa Martina, University of Genoa, Pansalis Pierre and Trainito Egidio from the Marine Protected Area of Tavolara Punta Coda Cavallo. So, in this study conducted between 2016 and 2020, we investigated the impacts of professional and sport fishing on benthic community, the presence of alien species, and the health status of protected species within the marine protected area of Tavolara Punta Coda Cavallo in the northeast coast of Sardinia. The marine protected area is divided into 3 zones with different degrees of protection. The zone A, the red zone, with the highest level of protection, the zone yellow, the zone B, medium protection, and here the zone C, in light blue, with the lowest level of protection. In the zone C, professional and sport fishing, navigation, and mooring are allowed. So, first, we define the fishing pressure inside the marine protected area. We have geolocated the professional fishing sites using GPS coordinates of signal buoys of fishing gears and reconstructing their tracks on this platform. As a second step, we analyze the impacts in the areas most subject to fishing pressure. In particular, we analyzed 82 sites using scuba diving photos and video transect, and we applied 3 indexes. Vulnerability is the combination of presence and density of erect species. I specify that the analysis of the impacts focus mainly on erect species as good indicators of mechanical damage. The second index, impacts, is the sum of 6 types of damages. Extirpation, which means the radical removal of erect species. Partial mechanical damage, which means breaking of branches and removal of part of chenenchyma. The presence of lost fishing gears, the presence of plastic debris, the presence of Caulerpa cylindracea is a alien species, and the presence of Phylograna salmacina complex as an indicator of thermal stress. The third index is the BC species, which means the presence of protected species under Barcelona Convention. So in the first map, you can see the geolocalization of the 86 cove sites collected. Most of them, exactly 83, are gillnets, in particular trammel and monofilament gillnets. You can see that the main fishing pressure is located here in the channel between the islands of Tavolara and Molara. In the second map, you can see the 82 diving sites investigated. And this is here, highlighted in light blue. It is important to say that all the sites are located within the zone C, which is the zone with the lowest level of protection. Recent studies have shown the presence in this area of a peculiar benthic community, characterized by the dominance of erect sponges, an irregular distribution of gorgonians, the presence of protected species such as Savalia savalia, and the presence of branches bryozoans. It has been shown that this habitat has a high naturalistic value. The next. This map illustrates the 3 indexes evaluated for each site. About vulnerability, we have given a score from 1 to 10 to define the degree of vulnerability of the site. A high score means a significant presence of erect species and therefore greater vulnerability. You can see that the 40% of sites show a score from 7 to 10, which means a high level of vulnerability. The next. About the other 2 indexes, impacts and the presence of protected species, we illustrate in this table the percentages of each impact and some representative protected species on the total number of sites analyzed. You can see that at least one impact is present in 99% of the sites. And here you can see the values for each impact. About the protected species, they are also present in 99% of the sites. In the picture, you can see Savalia savalia broken by mechanical impacts here. This is a colony of Savaglia extirpated and covered by epibionts. Paramuricea clavata, affected by warming in the distal part. And here you can see a colony dead and extirpated. Here there is Sarmacina filograna complex. And here a colony dead and extirpated. Eunicella verrucosa damaged at the base by a lost sporting line. Here there is Caulerpa cilindracea as a alien species, and a fishing line damaging Spongia officinalis here, and Axinella polypoides here. So the next slide, okay. In the study, we found that fishing activities act a significant impact on the communities living in the investigated areas. In particular, we found a strong fishing pressure in the Tavolara Channel where there is a habitat with a high naturalistic value. This area is in the Zone C, where there is the lowest level of protection. We found a significant presence of erect organisms in an evident state of suffering. The combination of fishing mechanical impacts and global warming increase the risk for erect species, favoring epibionts and alien species. So the recorded data support the need to review the previous estimates of vulnerability and usability of the area in order to develop management activities aiming at the protection and conservation of this unique habitat. Thank you for your attention.
Thank you, Laria. Is there any question? No. Okay, then I think thank you, Ilaria, and we will continue with the presentations. The next one is Eduardo Casoli, Impact, Remediation and Recovery of Coralligenous Reef after the Concordia ship wreckage.
Good morning to everyone. Thank you for introducing me. I would like to present the result and findings of 10 years of monitoring carried out by University of Rome La Sapienza in collaboration with the SIBM of Livorno in Italy. We are deeply concerned about the future of Coralligenous reef over the Mediterranean basin that are undermined by human pressure and climate change that probably will lead to the oversimplification of the assemblages. In the past 20 years, we focused on the identification of key and sensitive taxa to develop effective monitoring tools that improve our ability to detect impacts and quantify them. But the question is what we know about the ability to recover of this ecosystem. So just few studies have been carried out recently that highlight a poor and a lot of years to recover the function and the structure of Coralligenous assemblages. Today, I want to present an emblematic case, those of the ship wreckage of Costa Concordia, that we have monitored since 2012. Why choosing this case? because we know and we follow the evolution of the seabed at those sites. We fine scale mapping, we have a fine scale map of the impacts. We know, we knew the impacts that act synergically at each part of the seafloor. So the main objective of my brief presentation are to follow the 10 years dynamic of coralligenous assemblages at the study site to identify, to quantify and assess if the recovery of the reef have been achieved or not, and then to identify the taxa that are mostly responsible for the recolonization of the impacted reefs. To do that, we have to rebuild, we have to put together the pieces and what happened in the last 10 years. In 2012, in January 2012, the Concordia ship did a wreck at along the coast of Giglio Island. And after the wreckage, few months after the wreckage, in June 2012 and up to the end of July of 2014, the wreck removal phase was accomplished with a lot of engineeristic work to remove the huge ship. After the removal phase, from 2015 to 2018, we carried out an intensive and fine-scale remediation efforts with the aim to remove all the disturbances, for example, fine sediment that are spread all over the area, or man-made structures and debris that fall down from the wreck. So this phase eliminated all the disturbances created by the previous phase, the removal one. After the 2018, so from 2019, the area was kept close and is still close from further external pressure, like human pressure, I mean, like diving, fishing, and both activity to improve and to follow the natural recovery of the assemblages. So here in the map, we can show this distribution of coralligenous reef from 40 up to 80 meters depth. The reef are highlighted in purple, and they mostly colonize the two rocky ridges, the southern one and the northern one, in the left and the right side of the map that we monitored in 7 different sampling events from 2012, when the removal phase did not start yet, in 2013 and 2014, and then over continuously from 2018 and 2021. How we do that? We perform fixed survey along a predefined route, that are the route that we highlighted in red in the map, filming and covering the same route and filming at 1 meter above the substrate, so acquiring high quality video. Then from the video, we extracted at 8 fixed stations, the white dots on the map. At each station, we extracted 12 pictures from the video for quantitative and qualitative analysis of the assemblages. So we gather from each picture, we gather the following information: the presence of absence of conspicuous taxa, registered at the lowest possible taxonomical level, the presence of disturbance, such as debris, sediments, and the presence of base surface on the reef. Then we calculated the frequency of occurrence of each taxon. Then we calculated the alpha and beta diversity, the course index as modified by our group in a past publication of 2017 to cope with the future of the study site. Here's some results. We grouped together the information in the multifactorial analysis to improve the visibility. So we grouped together the information at taxonomical level. However, in the multifactorial plane, we can see the observation group in four different groups. The one constituted by 2012 observation when the reef were on a natural condition and were mostly characterized by green algae and octocorals. Then the samples of 2013 and 2014 shared the same position in the first quadrants and were characterized mostly by sediment and debris covering the reef. Then the 2018 samples were mostly colonized by bare surfaces due to the cleaning activities that were performed. And then the recolonization phase is highlighted by the proximity of the observation from 2019 and 2021 that share the same position in the factorial plane due to the increase in frequency of erect bryozoan, encrusting bryozoan, encrusting sponges, and erect ascidians and erect brown algae. Here, to summarize the evolution of to take a look and to give you a brief evolution, a brief visual evolution of the substrate. This is the situation at along the southern rock ridge at the 50-meter depth. This was the natural condition before the removal operation started. This was the situation of the 2014. So once the ship was towed here to Genoa, this is how the assemblages looks like now. This is the same evolution along the northern ridge, with the situation dominated mostly by those are mainly ascribed like a Coralligenous platform assemblages with an erect layer developed by erect green algae, and this is now the situation with the increase in abundance of erect bryozoan and hydrozoans too. So we recognize the we call the winning five, so the 5 taxa that mostly and rapidly colonize the bare surfaces. Reteporella grimaldi as first, then 2 or 3 maybe erect brown algae, the Carpomitra costata, the Arthocladia villosa, and Sporoctonus too. Antoine Constance showed high coverage on bare surface, and Alocinsia papillosa showed a high level of new recruits too. So to summarize our findings with other descriptors, the upper graph is relative to the frequency of bare surface of the reef that from the maximum recorded in 2018, so after the cleaning events, quickly decrease in the frequency of occurrence. So they are still present. This is an example. So when I talk about bare surfaces, I mean portion of bleached, totally uncolonized portion of bioconcretion. The alpha diversity as the mean number of species at each station increase since 2019 as the effect of new colonization of those bare surfaces. The beta diversity did not change significantly among years. I thought the maximum, the peak was registered in 2014 as an expression of scattered impacts and different magnitude impacts depending on the position of the station or the question. And the coarse index well described the integrity of the seafloor of the assemblages, with a slight increase in the last 3 years. However, the increase was mostly characterized, most of all in 2020 and 2021, by the presence and the dominance of erect bryozoan on the assemblages. So to conclude, the recolonization of the surface and the reefs impacted by by Concordia shipwreckage were quicker than we expected. So this is probably due to the remediation efforts that have been made and discovered and brought to light new surfaces available for biological colonization. The biological colonization, the recolonization of those reefs were mainly due to the settlement of opportunistic species that probably benefited of larval availability, absence of competition, and fast dynamics and growth, all characteristics that differed from the habitat former typical of Coralligenous reef. So now these findings open a new perspective, and we ask ourselves if this is a common pattern, and the winning five will lead recolonization and colonization of impacted reef over the world basin, or if it is a local pattern. However, the recovery of the assemblages and the recovery of Coralligenous reef is still far to be achieved, even after the restoration intervention, because the structure of the assemblages is deeply different from those we discovered in 2012. So even they spend a lot of time and cleaning the substrate, but not transplanting organism. The reef looks like very different from those was in natural condition. So we stress the urgent necessity to reduce the human pressure and to increase the restoration effort as both pressure removal and transplants of organisms of those reefs that are highly impacted, to maintain the structure and the functioning of Coralligenous reef over the whole basin. Thank you for your attention, and thank you for ISPRA and Genoa colleagues for the hospitality. Thank you very much.
Thank you, Eduardo. Questions? Yeah.
Thank you, Eduardo, for this interesting presentation. You know that yesterday Miche Bianchi stressed about the importance to investigate the complexity of the Coralligenous assemblages, considering all the layers, the upper layer, basal layer, and so on. So I highly appreciated that you used the coarse index in your evaluation that, as far as I know, is the only one today, is the only diagnostic tool today to evaluate distinctly the three layers. You showed us the overall result of the coarse index, but I wonder if you can, if you have noticed one or another layer that has been much more affected by this kind of physical disturbance, and in the recovery process, if there is a layer that is recovering faster than another one. Thank you.
Thank you for the question. Mostly the basal layer was most impacted. The disturbances were mostly cover of ice sediment over the area, and the physical impacts by rolling of debris that fall from the wreck. So the basal layer was mostly impacted, followed by the erect layer. So octocorals suffered a lot of the situation, and the one who recovered faster, or it seems, and benefit from the biological feature of the taxa that drive the recolonization was the intermediate layer. The bryozoan, the erect bryozoan, first of all the Reteporella species, and then the Pentapora and Adionella smithina too, quickly recolonize and quickly growth with big colonies, so increasing the descriptor, increasing the value of the intermediate layer.
we have over there.
Yeah, thank you for the presentation. It's a really interesting case study. And as you mentioned, there are a few studies about recovery. So the data are needed, and this is really nice study. I'm wondering if, because it's not clear to me, if you have data before because you show the pictures, but we don't have seen the data. So you started to study this place after the wreck or not? Or do you have information before?
Thank you for the question. No, unfortunately, we did not have information at this scale with this data and with this accuracy before 2012. This was one of the common dive sites, but they perform shallower dive, not from 45 to 70 meters. However, the 2012 samples were just before the starting of the removal activity. So the only disturbances was represented by the wreckage, by the ship. So no other disturbances acted during 2012. So we approximate to a natural condition, just not so close to except for the wreck.
Okay, and another comment that you mentioned also is that not the time needed to recover the structural complexity. And one of the things that you mentioned, but I think that it's to take in consideration, is that you will see in a poster presented by Yanis that we have seen that if you transplant the habitat former species, regarding, you can speed up the processes. So it's a situation, no? I think that with the restoration, we have to keep in mind that you should apply in local and specific cases, but probably it's a good place to see if transplantation or restoration action can speed up this recovery. So it could be nice too.
Yeah, definitely. We are performing transplantation, but shallower. Up to 40-meter depth. We are not able to work, and we are not allowed to work up to 70-meter depth. However, in the last 4 years, we transplant more than 400 colonies in the shallower Coralligenous reef that develop faster, that recovery faster if compared to the deeper one. But I definitely agree that a speed up, a kickstarter of recovery is the transplantation of those species that are extremely slow, that show extremely slow dynamics, like octocorals. And probably they would stop the bryozoan and other and hydrozoan colonization by the competition, probably.
Okay, thank you.
Yasin, please, there. Udures.
Thank you. Very interesting indeed. You want to help restoration. Why don't you let nature be natural?
Thank you for the question. We just remove the disturbances at those depths. I mean, between 40 and 70 meters, we just remove all the disturbances. I mean, fine sediments and debris that are alloctonous stuff, and then the nature is doing the work, because we are just observing the recolonization. So the results that I have presented are only natural recovery of the reef, but a clean reef. So the unique case is represented by we have the opportunity, by the fact that we had the opportunity to remove the pressure and the disturbances and the impacts as well. So the nature of the assemblages are recovering naturally without any transplantation intervention at the depth comprised between 40 to 70 meters. Thank you.
Any other question? No. Okay.
Thank you very much.
Okay, so now we move to Marmara Sea, soft corals and gorgonians. Okay, we have a new red Utopku.
Good morning, everyone. I will present about the soft coral and gorgonian assemblages. They are exempted of thermal stress, but poor things are threatened by high anthropogenic pressures in the Sea of Marmara. I will start by briefly introducing Marmara because you may not be familiar with it. It connects two seas with really different oceanographic conditions, the Black Sea and the Mediterranean Sea, with only two very narrow straits. And because the waters in the upper layer originate from the Black Sea, it's very brackish light. And of course, it's separated from the lower layer, which is more saline because it's coming from the Aegean, the Mediterranean Sea, with a permanent halocline. It's there always year round. So in the lower layer, we have 15 degrees Celsius year round all the time. It's 38-40 salinity, and we don't have major seasonal variations because the Black Sea layer is highly turbid. It's quite dark, dim light conditions, we can say. So it's kind of mimicking the mesophotic conditions like 50 meters and deeper in the Mediterranean. And we have three main types of alcyonarian assemblages. We can have these dense yellow gorgonian assemblages. They can eventually sometimes have some Spinimyrcea clavereni and Paramyrcea macrospina among them. They are really very beautiful. Another assemblage is formed mainly on detritic bottoms. The coral gardens of Paramyrcea macrospina, Spinimyrcea clavereni, and Paralcyonium spinulosum. I'm sorry, I'm a terrible photographer, but I really don't have time underwater for nice pictures, and I have a poor camera. So sometimes rich divers join me voluntarily, and I can have nicer pictures. The third type is these coral gardens with Paramyrcea clavata, and of course they are accompanied by Paralcyonium spinosum sometimes. On this one, you can see some macrospinas or cavolini occasionally. Oops. Okay, that looks like a safe haven for them. No thermal stress, no heat waves or so. But unfortunately, it's not. This is more like a war zone because we have these rising pressures since 1980s. Of course, you see this ugly picture of poor Istanbul city, which witnessed a rapid population growth, all the Marmara region, but particularly Istanbul, of course. Also, there was an industrial revolution in the surrounding region, again, started in 1980s, particularly on the southern coast of the Marmara. And you all know that that also coincides with the catastrophic degradation period in the Black Sea. And in the same period, the government sustained industrial fisheries. They had subventions and so on, and they started to reign over small-scale fisheries, which were the only type of fisheries until then. So everything started to go bad since then. And recently,
we had these, I'm mainly working on the northwestern part of the Marmara, namely Prince Islands region. And in this group of islands, we had 2 uninhabited islands until 2015. And then at that time, they decided that why leaving these islands empty, and made constructions on it. Of course, this resulted in excessive sedimentation and covered all my poor Gorgonians. Several colonies died very quickly during that time, especially those that were close to that island. We have this very iconic picture, which shows just with 1 year separation of the same colonies. We have this beautiful Paramucea clavata at the center, and just next to it, we have the beautiful Spinomucea claverini, and just behind claverini, we have a tiny macrospina, Paramucea macrospina. And in the 2015 picture, you can see that the Spinomucea claverini skeleton is gone very rapidly. It's much more, I don't want to say soft, but it's less strong than Paramucea clavata skeleton, for example. So it's in general the first to leave the bottom, but clavata, of course, is still there with its skeleton. And you can also see behind, I don't know if the, well, you can see here the, no, that's fine. Just, I didn't see if the icon is there. Macrospina skeleton is also still there. Then last year, we had this huge mucilage phenomena. We have a poster on it, and Cansu will present you some videos and aerial photos of the mucilage. It was really terrible. It started in 2020, you know, this fabulous year, 2020, with everything going great. And we had this huge mucilage that started in autumn, and it reached its highest level in spring 2021, and it lasted until summer. And it covered everywhere. They were really huge, larger parts than a human, for example. It was really scary diving during that time, and it covered everything, of course. So what we do in Prince Island to study on ocean areas, we have these 9 sites that are regularly monitored for the distribution and density. And we recently studied the demographic pattern of the yellow gorgonian at 2 sites. We also studied the population genetics of the yellow gorgonian in 4 sites in Marmara and 7 sites in the Aegean Sea. And also recently we just, actually after the sedimentation, we found out some opportunistic bacteria and the fungi on the gorgonians and soft corals. So we said, okay, we need to look for the bacterial microbiomes, and we studied this by 16th rRNA genotyping. And what happened was really sharp decreases. It started with the excessive sedimentation, with the constructions and so on the uninhabited islands. We almost lost everything, mainly soft corals, which decreased by 92% almost. And then with the mucilage, of course, it became even worse. Now, imagine we have these hundreds of Paracionium spinosum, for example, several hundreds of Paracionium spinosum, which suddenly decreased. And now we have only 4 or 5 sites that you can still see them more abundant, even though it's not, of course, like before. And for the gorgonians, during the excessive sedimentation, we lost 88%, except for the yellow, because they are very dense at some sites. So I wanted to exclude it and deal with it differently. For the others, so Macrospina, Spinomucea claverini, and Paramucea clavata, we had these sharp decreases. And there were a lot of dead skeletons everywhere. With mucilage, actually, gorgonians dealt better than soft corals. For example, with hydrozoans, during the excessive sedimentation, opportunistic hydrozoans have highly increased. With the mucilage, it was just the contrary. The hydrozoans also decreased a lot. This is mainly, or probably I can say, mucilage is harder, more heavy, and it covers everything like a blanket. So even the hydrozoans were dead after the sedimentation, but gorgonias, like an umbrella, they dealt a little better than the soft corals and hydrozoans. So for the yellow gorgonian, unfortunately, we don't have data previous to the excessive sedimentation, but it was this one in 2016 is just after the excessive sedimentation. So I think we can imagine that these necrosed and dead colonies were probably healthy before the event. And you can see that it's more or less well-structured population because it has a high number of small colonies with the presence of large sizes together. But after the sedimentation, after the mucilage, you can see that there is a shift in the size frequency distribution. Now it's dominated by very small, around 5 centimeter colonies. These populations, of course, they were already impacted by the excessive sedimentation, and then they had this mucilage. I think this is probably due to the umbrella effect of the large sizes who died. Mainly they were the ones to die with the mucilage. And then we had, of course, after the sedimentation and mucilage, more space available for new recruits to settle. For the genetic connectivity, we had 3 sites in the northern and 1 site at the south and 7 others in the Aegean. And the structure gave us 2 main groups that clustered regionally, unsurprisingly. And actually the ones in the Marmara were the ones that had the lowest genetic diversity and richness. And imagine that now they are decreasing, their numbers are decreasing. They are dominated by young colonies, so this might have some effects in the near future on the reproduction and recruitment. So I'm afraid these populations might be at a risk of bottleneck in following years. For the bacterial microbiomes, we had three alcyonarians, Alcyone macaulay, Anisella cavolini, Paramucea clavata, and another octocolor, Veratilium cinnamorium, and the hexacoral, Savalia savaglia. And they had dynamic but distinguishable bacterial communities. Actually, the host identity explained only 30% of this variation. Probably the environmental factors were more important in this variation than host identity. The gorgonians had in their core microbiome endozoicomonas bacteria, like everywhere else previously reported in the Mediterranean, but they were much less abundant in the Marmara, probably again due to anthropogenic stresses which caused shifts and changes in the microbiome composition. So what's the situation now in Marmara? After the mucilage, the ministry realized that there's a problem in the sea, and they prepared an action plan to reduce pollution and increase water quality. And then there was this consecutive integrated strategic plan. They also decided to declare the whole Marmara as a special environmental protection area. It's been more than a year now, and not much has changed, I can say, in this sense. Fisheries, for example, industrial fisheries continue as previously. And there was just before I came here a new regulation, a ban to collect gorgonians. Well, they were not much collected anyway, but of course they come along with the porcine nets. So this is a major problem. Maybe it can have some, I don't know, preventing effect on this one. And we had this small, very small MPA. I am more happy with this one than the large MPA, because this was actually following our transplantation efforts, thanks to help and support from Kim. We had, you know, I said 2 islands open to constructions. One of them started, the other one was planned to start in 2019, but thanks to a lot of support from locals, people, scientists, and etc. We all joined, and we actually won this war, and the construction was cancelled in Syria. But of course, as a precaution, we have already started to carry some yellow gorgonian colonies to 13 kilometers away, just in case there might be constructions. So they are still there. We have some clones so this is the island that was supposed to be constructed, and we carried them here. And as they were doing quite fine, the ministry understood our efforts, and they declared a small MPA. Just before the status, actually fishing nets, there were high fishing efforts there, and purse seine nets unfortunately took some of our transplants, but otherwise they're doing fine. They were really small, around 10 centimeters, and now they have quite grown up and in 3D dimensions. So we are quite happy with them. So what's next? We will of course continue on monitoring the distribution density and demographics, and we started a new study to study the reproduction pattern of the yellow gorgonian in Marmara, because there's this seasonal pattern in the northwestern Mediterranean, but how does it deal with the Marmara conditions, which just don't show much major changes, seasonal changes. So we will try to see that, because we already studied previously Spinimyrcea claverini, and it was active year-round. So we don't know, and we will try to see that. We can also consider some restorative measures, maybe if we see that the action plan and the MPA status are having some positive effects on the sea and the conditions are improving, then we can eventually consider such measures. And I would like to thank my team and to you all for listening. Thank you.
Thank you, Nur. We have time for one question. Yes, Laura.
Thank you, Eda. It's a really nice presentation, and the case of Marmara is really interesting. It's a pity, no, the situation.
Yeah.
The problem of that, no, of this place is the lack of enforcement, no, because as you mentioned, no, there are because this happens in all places, I think, no, in all marine protected areas, but especially in this case, no, what is the confidence that these regulations can be implemented in a real way to act or to develop restoration measures? You have to be sure that the stressors are not there. So
yeah,
I'm not sure that the case of Marmara, no?
Yeah,
what do you
my
think?
confidence is none, actually. But I think we need more public awareness. I'm also in the NGO. I'm working hard for that part also. People, when you tell them about gorgonian corals in the Marmara, just there in front of you, a few kilometers away and so on, they are really surprised and excited. And during the mucilage, we really worked hard. That was the result, actually. The action plans was the result of the public reaction. But then, of course, people forget things. Now, this year, we didn't have a mucilage, probably because we had a very cold winter. So no mucilage, and no mucilage, no one cares. The sea looks blue and fine, so they just forget about it. But of course, we will keep trying. We need more NGOs, actually.
Sure. Thank you, Eda.
Okay. Just one question. So, oh, sorry, one more.
Just following the question. So I have the impression, working with MPAs, assisting to the session yesterday about Posidonia and today about Coralligenous. So we did our duty as scientists. It is true we have to keep educating population, etc. But I guess we have to start publishing the fact that the authorities don't do their duty. The law exists. So there is plenty of people that is interested in protecting the environment. We see the movement of young people, etc. But I guess what is missing is the presence of the state, the law, and the application of the law and the enforcement of the law.
I agree. What can I say?
Otherwise, I guess we don't. So we will meet again in 4 years here and there, saying always the same thing.
I keep actually writing about it. I write in Turkish because I know they won't read it in English, but let's just be hopeful. Thank you.
Okay, thank you, Nur. Okay, so now I think we have Carlos online, Carlos Jimenez. No?
It's not, it's,
Oh,
I'm
Madeleine?
presenting for Carlos, yes.
Okay, then is it online?
Yes,
Okay, could you share your presentation?
of course. Can you see it?
Yeah, but it's not in full screen.
How about now?
Okay. Perfect.
Okay. Should I start?
No. Go on.
So hi everyone, my name is Magdalene, and today my presentation will be focused on the effects on the destruction of Coralligenous concentrations and other benthic zoophilic communities in the MPA Cape Greco. Sorry, 1 second, let me fix this. So I'll start by talking about how disturbance can shape communities. So if we look on the left side on the graph, at the bottom of the graph, we have the disturbance, and then on the left side of the graph, we have diversity. So starting with A, this is where we have the lowest disturbance, but also very low diversity. This is because in an ecosystem where there's a very little disturbance, only the most competitive individuals will dominate, excluding the less competitive species. Moving to the peak of the graph, where we have intermediate disturbance, the intermediate disturbance creates multiple niche for different organisms to occupy at the same time. Therefore, we have high species coexistence and high diversity. Then moving towards the right side of the graph, where we have the highest disturbance, but also the lowest diversity. This is because at this point, the environmental stressors are too high, and therefore the most resistant species are the only ones that can survive and dominate. Now going into a brief history of the Cape Greco, the 2 important dates that you need to know is that in 2004, it became a Natura 2000 site, and then in 2018, it became an MPA with zonation. The Cape Greco occupies an area of 2,003 hectares, out of which 54% of it is terrestrial and 46% of it is marine environment. It is mostly rocky with steep slopes. It has several caves above and below sea level. It has Posidonia seagrass in the shallow zones. It also has sandbanks and reefs. It also houses several, why is it moving? It also houses several important species, from marine mammals to plants to calcareous algae to invertebrates. And it also homes several critically endangered species like the Mediterranean monk seal, Monachus monachus. As I mentioned, the Cape Greco has 3 zones. We have the core zone, which all activities are allowed except any type of fishing, whether that's commercial or recreational. We have zone 2, where all activities are allowed except recreational fishing, and then zone 3, where all activities are allowed. Now, this is a legend of the map that we'll be seeing next. Starting from the left side, we have diving, swimming, and research. And then on the right, we have the icons for commercial fishing, recreational fishing, and education. So Cape Greco is located on the southern east coast of Cyprus. And in red, you can see the core zone where neither commercial fishing nor recreational fishing is allowed. Then we have the 2 yellow zones where recreational fishing is prohibited, and we also have the blue zone where all activities are allowed. So what happened? In early February of 2021, there was a really bad storm, which forced quite a big ship that was 125 meters in length to seek shelter in the MPA and anchored itself in the zone 2 of the MPA. The depth which it anchored was between 23 to 27 meters, and because of the size of the ship and the weather condition, it actually had to use 2 anchors to hold itself in position. So if you look at the asterisk, this is where the ship anchored itself in the MPA. Once the storm passed, the ship started to lift its anchors, and this left behind meter-long scars on the seabed. It also dislodged more than 30 large boulders, or actually some of them were broken off, and the largest one we measured was about 12 meters cubed. These exposed Sciaphilic communities originally thrived below the large boulders directly to light. At this point, we didn't know about the ship in the MPA, but we were pretty fortunate for two reasons. The first reason is that the area has been monitored sequentially since 2016 as part of the Protomedea project. And we also had a monitoring scheduled about 2 to 3 days after the incident happened in the MPA as part of a different project known as Life IP. The Protomedea and Life IP projects use a similar method of quantifying benthic communities, where they use a systematic way of photo quadrats to quantify it. And the first survey we have is of June of 2016, which is what we will be using in this presentation as a baseline survey. Then for the destruction, we use a similar method to quantify the benthic community, where we laid down transects that followed the contouring of the boulders. We did 3 surveys. Our first survey was February of 2021, which is the comparison we'll be using today. But we also did 2 other surveys in April of 2021 and July of 2021. So we used photo quadrats to quantify the benthic community and then measure the percentage cover using photo quad. The species identified were then placed into 9 different categories, from substrate, calcareous algae, microalgae, bryozoans, cyanobacteria, sessile mollusks, porifera, corals, and other organisms. The first statistics we did was diversity index. So if we looked on the left side of the slides, where we have the Shannon diversity index indicated by the purple line, on the left side we have the index, and then at the bottom of the graph we have the 2 time periods. So we can see that there's a dramatic increase in the community from July of 2016 to the February 21 sample. Now, the diversity index is made up of the species richness and the pillar's evenness. So if we move to the top right corner where we have the richness, we can also see that there's an increase in species presence between July of 2016 or 21, or actually we can see more species. Then at the bottom right corner, we have the pillar's evenness. So for those that don't know, a value of 0 means that the community is dominated by a single or few species, while a value of 1 means that there is more community changes from dominant to quite a diverse community. So if you look at the diagram at the bottom, we have the anchor moving along the seabed as it's being lifted up. It gets stuck in the continued moving. Since there were no clear signs of necrosis during our initial survey, this suggests that the destruction occurred only a few days before our visit. Also, the exposed Sciaphilic communities create this unnatural increase in epibenthic diversity. Then we ran non-metric dimensional scalings, then MDS and Adonis analysis. So if we look at the graph, the blue cluster is our sampling from the 2021 samples, and then the orange cluster is the 2016 samples. So both of the NMDS and the Adonis show that there is this separation between the 2 communities from our samples. The main species from the 2016 samples were macroalgae and calcareous algae, while the main groups in 2021 were bryozoans, corals, porifera, cyanobacteria, and mollusks. Also, 2 of the species identified in the 2021 samples were Madrasis farancenis, which you can see on the left picture, which is a coral, and the Celeborina caminata, the bryozoan on the right photo, which are known Sciaphilic species, and they're also very rare or almost never found on flat surfaces exposed directly to light, as we found them in this case. Also, many of the species identified in the 2021 samples are more cryptic and abundant in semi-dark habitats compared to the ones in the 2016. So the Sciaphilic communities that were identified on the underside of the boulders that were flipped over are of unknown age, and these communities are also considered important repositories of diversity in any community. So just a quick summary of what happened. due to the bad weather, the storm, this forced a ship of 125 meters to anchor itself in the Cape Grego MPA Zone 2. While the storm passed, the ship tried to lift up its anchors, dislodging several boulders, exposing in this way the Sarfili communities underneath directly to light. This increased the biodiversity of the area, which was quite unnatural and also was short-term. The community composition between 2016 and 2021 were significantly different, and the main species for this difference were macroalgae, calcareous algae, cyanobacteria, mollusks, porifera, bryozoans, and corals. I will also give you a brief insight, a quick tip into how things progress in our next surveys. So basically what happened, subsequent surveys showed sequential widespread mortality and necrosis of organisms, which was expected. Since these species are usually confined in dark habitats, Since they became exposed directly to light, the mortality would increase either due to prolonged periods of exposure to light or by predation by biological agents. Yes, and thank you for listening, and to all my colleagues. Any questions?
Thank you, Magdalena. Do we have questions? If not, do we have another presentation from yesterday? Yeah. Well, okay, thank you, Magdalena, for your presentation. And now we have from yesterday one presentation online that is left. It is Marina. Do you hear us?
Hello. Yes, I'm here.
Marina.
Can you hear me? Can you hear me?
Well, we can hear you, but not very well.
Oh,
could you share your screen?
Yeah. Can you hear me? Can you see my screen?
Yes.
But I can try to disconnect my headphones if it is a problem, to try if you can listen me better.
I can hear you.
Okay, so I will start. Thank you very much for all the attendance. I will now present our common work, Can an emblematic species become a pest? The case of Astrospartus mediterraneus, Echinodermata fibroidea, in the artisanal fishing grounds of the Cap de Creus area in the northwestern Mediterranean Sea. First of all, unfortunately, our colleague Marina that was the presenter, has not been able to connect. So I'm presenting, I am Yanire. And I would also like to thank the organization for the opportunity of the reschedule. Our species is Astrospartus mediterraneus. It's the only gorgoncephalid in the Mediterranean Sea. It is a suspension feeding of urate with nocturnal feeding on zooplankton and suspended particulate organic matter. And its distribution is from eastern North Atlantic coast of Africa and Europe into the Mediterranean Sea. It is considered a rare and emblematic species in need for conservation and protection. There is a scarce information about its biology or ecology, and it has been out of the scientific community scope for years. Also, for years, its presence had been known by local fishers and scuba divers in the study area, yet scientific records were rare and corresponded to isolated individuals. The study is framed in the MITICAP project. This project is in collaboration with scientists and fishermen, specifically artisanal fishers from Port de la Selva and Cadaqués in Catalonia. They noticed an increase in Astrospartus mediterraneus over the past 5 years, and this species conform a huge part of the bycatch. The fish's perception is of a problem, so we conducted this study. Why it is a problem? Because the morphology of Astrospartus provokes that they are entangled on the fishing gear. meaning an extra time in cleaning and mending the gear. The study area is located in the Cap de Creus, that is a maritime terrestrial natural park in 1998, then a site of community importance and a special protection area in the Natura 2000 network. This point is the most westerly point of the Gulf of Lions, and the fishing grounds of the study collected data from fishing events in yellow dots and with video transects in red. So they are shared by collaborating guilds of Port de la Selva and Cadaqués. The area is characterized by a complex bathymetry. There is an area dominated by submarine canyons, and it means a complex bathymetry with high slopes. The fishing samplings where the data from fishing samplings was collected by a total of 143 fishing samplings. The scientists joined the artisanal fishers fleet in regular fishing journeys, and in total they joined 9 vessels, 6 vessels from the Port de la Selva and 3 from Cadaqués, from April to August of 2018. Data from different fishing gears was collected, and we checked the presence of the species Astrospartus mediterraneus by bycatch. The pictures were taken with a grid, and the perimeter and the diameter of the central disc was measured. Regarding video recording, a total of 19 video transects were collected by means of remote operated vehicle, having data for a total of 9 kilometers with a depth from 11 to 18 meters water depth. The sampling units for the analysis was considered 2 square meters. That is the measure commonly used for this kind of studies, and the analysis were carried out with Final Cut Pro 7 video processing software. The information compiled included depth, associated substrate type, geographical coordinates, and slope, and also we collected data from the perception of the fishermen. We collected the technological ecological knowledge, and we not only collected the perspective and the impression from the fishermen of the study area, but also along the coast of Catalonia. And in this survey, we collected information about the fishing history, the gear use, among others. Regarding the results, we observed a diameter size of 267 centimeters in mean. And it's important to highlight the asymmetric distribution with a dominance of small and medium size classes. Also, with the kurtosis test, we observe significant thickness with size prevalence of 2 to 3 centimeters, individuals of 2 to 3 centimeters. Regarding the geographic distribution, from the different fishing events, we can say that there was a uniform distribution of the individuals regarding abundance. And then we observe in the video transects 2 main aggregation zones, one in the central north of the Cap de Creus and another in the Massa d'Oros island. That these areas coincide with intense fishing activity there. Regarding the bathymetric distribution, we obtain a distribution between 35 to 85 meters, with a noticeable increase in the continental shelf below 50 meters. And it's important also to mention that, related with the different fishing catch, the bycatch was shown almost exclusively on trammel nets. Related with the habitat preference, we observe in order a preference for rocky substrates, then with the presence of ecosystem engineers, specifically Gorgonian forests, and then with sloping areas. And related to the fissures survey analysis, we observe clear differences among the south region and the northern region, and with number of individuals called ranging in the areas with presence from 10 to 30 individuals. Also, in some areas there were observed more than 50 individuals caught per fishing event. And related with the time invested in cleaning the fishing gear, the fishers express their concern because they spend more than 13 minutes, in some cases more than 5 hours, in the fishing events. So the mean time spent for fishing the events derived from this outcome, outbreak of Astrospartus, is among 1 to 3 hours additional. So then, related with the population structures, we obtain that 70% of the individuals range from 1 to 3 centimeters of this diameter, which means that we are in front of a small a young population in comparison with other populations. For example, in the literature we have the registers of 79 centimeters in the north of I think that it's in the north of Africa, with and it means that in the categories aggregations are young individuals, and we are in front of incoming outbreak, a coming outbreak. So we also observe, which seems to be parental caring in some individuals. That was already reported with other species of uroid species. Related the mass occurrence, we are in front of an outbreak because there is a clear density growth in comparison with other Mediterranean areas, and we think that it could be maybe by food source increment, by water eutrophication, increased water temperature, or rain reef nutrient enhancement, due to maybe the temperature increase that amplifies and enhances reproductive period and reproduction output respectively. And also maybe it can be due to overfishing of predators, for example, of the mollusk caronian. And so a clear perception of the fishers as perceiving the species as negative was collected. It means an economic loss and an obstacle to job performance in an already difficult, the artisanal fishers are a difficult sector, So we collected, well, we mentioned more, in some cases more than 5 hours, and it means in economic expenses, it means from 30 to 100 euros per fishing event, which means that we should be investing more to repair the damaged fishing gears or offer protection and measures for this kind of activity. And other solutions are modify fishing habits, sharing habitat with target species. The ecological traits and habitat preference of Astrospartus mediterraneus aggregation determined in the categories marinaria, where sloping rocky substrates with presence of gorgonians with a water depth range from 50 to 80 meters. We have noticed aggregations of young individuals. those are the most dense aggregations in the Mediterranean Sea to date, and the fishers' perception unanimously is that Astrospartus mediterraneus, it is a handicap in terms of monetary and time losses. New ecological insights and fishers' perspective, still there is a massive gap in knowledge regarding the biology of Astrospartus mediterraneus, and so the reasons for outbreak could not be determined. So additional biological studies are further needed to improve our general understanding, and also for preventing it from becoming harmful for both the ecosystem and the fishes. And due to the environmental and anthropic changes on a local and global scales, we think that phenomena like this will be on the rise. This is the bibliography use, sorry. And I put here the email of Marina, the corresponding author. So yeah, we can, if anybody has a question, but I am not able to answer. Marina could, of course, be reached, and you can address the questions to her too. Thank you very much.
Thank you, Marina. Well, we managed to finish also this presentation from yesterday. And now if anyone have a question for Marina, or maybe we can take a general discussion, 5 minutes, not more. Well, we were speaking about different impacts and disturbances. Recovery maybe is the coffee break that everybody wants, but maybe if you have any general comments on presentation from this morning.
I want to mention that this is Paolo said also, and we can see in all the in most of the presentations, not the need of marine protected areas, but the reality is the lack of enforcement of them. So this is a thing that we should show, as Paolo mentioned, in the scientific literature, but also in the forums, because we are talking about marine protected areas, create more marine protected areas, but it's not a good tool if we could not implement the rules at the management tool. So I think that it's a really big question for Coralligenous conservation, and I think that we should take into consideration. And on the other hand, it's linked to Kim's talk about the implementation of these scientific surveys in marine protected areas. But we should also realize that in some marine protected areas, there is a nice staff working on these surveys. But on the other hand, we have a lot of marine protected areas without the staff, and it's the scientific teams that do whatever they can. So this is another difficulty so we need fundings for all of these challenges.
Thank you. I just want to get the point by Paolo, because it's true that we have been meeting in these several nice symposiums and the MPA forum, and the situation is not good. I mean, the evaluation, the assessment is that we are not going in the right trajectory. And I had the opportunity to be in an event these last weeks, and there was a session about the legal tools that the environment fighters, I would say, or the climate fighters have. And there is an interesting movement that there are some lawyers, in fact, teams of lawyers that are working to put lawsuits against the states for not achieving or doing enough to reach the Paris Agreement goals, for instance. And they won. And now, for instance, in Netherlands, there was a lawsuit like this, And now the government has to put many resources and make big changes in the country to meet the Paris Agreement. And I think, and I asked, but it was run out of time with the lawyer, but I wanted to contact her because I think that we can do the same for the goals on protection, because we have this international agreement on at the European and at the Barcelona Convention agreement on the percentage of protection, but not only on the percentage of protection. We have the agreement, it says as well that it has to be effectively manage the MPA. So this joins all the problems that we are seeing here. And probably, as I say before, that we need any kind of expertise because the challenges are really high. Maybe we should move and start contacting those lawyers and start filing lawsuits against the countries to reach this protection an effective way. Thank you.
Well, sooner or later we come to the money or economy. And I think that we are, as scientists, we are offering some network and some examples of how it is good working or bad working and what we can do, what we in the good management MPAs we can change or maybe not. But on the end, I think also that we should not only contact lawyers, but also economists to put some more numbers value of the nature, because when we talk with the decision makers, except we speak about the money and the suits, I think they simply don't understand. And sometimes they understand, but they pretend not to. So maybe it's the good way to put the pressure on, because we know impact assessment is something that should be done, but like in Marmara, the work that is going on is allowed. So something is not working properly. Any other comment? Yes, please. Madi? Director.
Thank you. This is not, it's personal insights. Just to react to what has been suggested. As far as I know, not most, but many of the numbers on which states have engaged and commit are, for example, for the 30% protection or similar things. It's regional, it's not by state. So sometimes it's not that easy, and you enter into very complex regulatory details. Of course, lawyers know more than we do, but I'm not very excited to tackle the problem from that angle, because vis-a-vis the biodiversity protection in general and marine biodiversity, I agree with Vesna on the psychological preparation via economists and specialists in transforming our language, because we don't speak the same language as the decision makers, unfortunately. And we need to transform our language and our numbers into more audible and understandable numbers for the decision makers, because I'm sure if at the right level, we have these messages very clear, and what are the dangers and the risk we are taking by business as usual, I'm sure that there will be action. So I don't know the steps and layers of transformation of our language in order to transform it and make it more touching the point for the decision makers. Thank you.
Okay, thank you. Maybe if there is no other questions, we can go for a coffee break and 10 or 15 minutes after what you said.
10
10
minutes.
minutes. Okay, thank you.
10 minutes. Yes, we have a poster session with 20 poster to present. So thank you.
Similar. You may too. Coralligenous are under various source pressures. Under not to mention all these. Yeah, but then it's missing, yeah. I think, but also I think it's more condensed, it's better.
I call Aldo to present the MAVA seabed habitat, a complete repository of standards, habitat and composite maps. Thank you. Yeah. It should be, sorry. Yeah.
And, good morning, and thank you for allowing me to present the poster in this session. And our poster is about the emergent seabed habitat.
Sorry, colleagues. Sylvia.
No,
no. Wait for them to see.
Okay, so the poster is about the emergent seabed habitat portal, where you can freely access to a huge amount of seabed habitat spatial data, standardized and ISPRA compliant. Since the first phase, the main product of this portal was the broad-scale modeled habitat map that initially covered just the Western Med and the Northeast Atlantic, but now encompass all the European seas, covering about 11 million of square kilometers. And it represents probably the unique example of model of pan-European seabed habitat model. And but throughout the different project phases, this main product was complemented with other spatial data, in particular over 900 habitat maps from survey, and about 500,000 habitat point data, and more or less 100 single habitat model. Furthermore, the project is collaborating with the Regional Sea Convention, in this way taking into account a specific request. For example, at the Mediterranean level, we were asked by SPARAC for the construction of a composite map about marine vegetation, and where possible also Coralligenous and all the bioconcretion. These products are now fully in place and available on the portal, providing in this way a useful tool for the assessment of these key habitats. If you are interested in this initiative, you can visit the poster and ask me for more details. Thank you very much.
Thank you, Aldo. Now we have the poster concerning the structure of Coralligenous community of Avelik Island Natural Park.
Thank you. Hello, dear colleague. Sorry. I think it's okay now. Our studies the structure of the Coralligenous communities in a natural park in Ayvalık area. With this study, of course, everybody knows the importance of the Coralligenous habitats, but unfortunately, this habitat is under some threat, like overfishing, excessive recreational diving, invasive species, mucilage, and especially overfishing in the area. Ayvalık Islands Marine Park, in actual nature park, there's a place of the huge area, and most of the area is part of sea and some islands, and we choose for station. 2 is with gorgons, we call G station with dots in red mark, and 2 is non-gorgonian station, and G which is dot by yellow mark. And we assessed the Snelle Cavalloni gorgonian population in Ayvalık Islands area. For this aim, we take some samples with photoquadrat analysis, and also some samples for certain identification of species, especially for sponge and Coralligenous. And we found some results, and my colleague Tancred will present our results and discussion. Thank you.
Thank you very much. No, it's not math. It's not over yet, sorry. I should have. So now let's proceed to the results. Overall, in 4 stations, 61 benthic taxa were reported. When we take into consideration the presence and the absence of gorgonians, we have been able to observe that in the station with gorgonians, the most present or benthic coverage taxon was sponges, followed by chryophytes, and at last rhodophytes. In a reverse trend in the station without gorgonians, we have observed that rhodophytes were the most dominant, followed by chryophytes and sponges. And in summary, we can say that in a station with gorgonians, 47 benthic species were assessed, while in the other categories, 50 species were reported. Our results are in accordance to the other studies that have been performed in the agency, and we have been able to observe the significant statistical differences between singular station, Ayvalık Taşları, and Güneş Adası. So, in conclusion, what can we say? The gorgonians are architect species, especially Unicella cavallini, is able to provide greater biodiversity enhancements. It is stressed and in risk of disappearing because of algal turf coverage of fishing nets, but also of invasive species such as Colerpa racemosa. And at last, we have a management plan for the Ayvalık MPA, which has been approved since 2004, but until now, no measures to enhance the application and to promote the direct protection was undertaken by the government. So there is a lot of modification, updates to apply to our management plans. Thanks to our scientific team who also greatly contributed to the work, but also to you for your attention and to the SPARAC for the organization of this symposium.
Okay, thank you. Now I call Patrick to talk about the ecosystem-based index to assess the ecological status of Mediterranean coastal detrital bottoms.
Thank you, Atef. Okay, so I will present you a work about the definition of an original index based on ecosystem-based approach, once again, but this time on coastal detrital bottoms. So it's the main interest of this work is it's the opportunity to highlight the importance of this very interesting habitats, widespread with a lot of interest talking about functioning and ecosystem services. So to catch your attention, maybe I can start with the name of the project when we do this work, the project ACDC for the assessment of coastal detrital conservation status and ecosystem-based approach. So I'm quite proud of this name. So why not a working name for a sandy bottom project? So, and most of the time, coastal detrital bottoms are known for the association with rhodolite beds, merged beds, and main studies in the past are based on this association or the taxonomy on the fauna of the sediments. But there is a macro fauna like macroechinoderms, cnidarians, fish assemblages that are of very important interest in terms of ecosystem functioning. So it's a work done by Gis Posidoni and the Aix-Marseille University, the Mediterranean Institute of Oceanology, and granted by the French Biodiversity Office. So the challenge for the poster is to do the synthesis in only a poster of 200 pages technical report. The different steps were like a workshop gathering 15 experts on benthic ecology to define a conceptual model of the ecosystem, to define the suitable descriptors to assess this compartment. And after this important stage, we implemented on the field both with scuba diving at the upper zone between 30-45 meters, and with a DIY device, the BioCube, using GoPro videos and a time-lapse camera to take actual photo quadrats. So we have some results resumed in the poster, so I'll be glad to exchange, to share it with you. So thank you for your attention.
Thank you, Patrick. And now I call Ignacio to talk about mapping community interest habitat in the Columbretes archipelago.
Right. Good morning, everyone. My name is Ignacio Baena, and I'm here to talk to you about the research we've been doing lately in the Columbretes archipelago. I'm here as a part of the Oceanographic Center of the Balearic Islands, and this project has been done in collaboration with many research oceanographic centers of Spain. And as a bit of background, the Columbretes archipelago is a teeny tiny group of islands located in between the Balearic archipelago and the closest port at 30 nautical miles, roughly. And here we have the previous cartography that we had. This one is from the 80s and was developed by means of ROV service as well as dredge sampling. And here we have another map that was focused on specific spots of the archipelago, developed by Cristina and colleagues from Barcelona and the Balearic Islands as well. And as part of the Intemares project, the idea was to do the map covering the whole SCI site, which was, by the way, declared in 2006. Here we have the different transects developed on around the protected site. And we did some scuba diving transects, ROV. We also did dredge sampling. And with the carousel wattle sampler, we can see a lot of concentration down here. And this is because this is an area where there are gas emissions that produce an acidification of the water. Cristina also published a paper in 2015, if you want more information about that. And some pictures about the reef habitats that we found around the protected site. The multi-beam echo sounder allowed us to link the habitat identification with the environmental variables, which were mainly depth and derivate variables, as well as the reflectivity of backscatter. And this produced the final habitat map. We can see on the left side the complete map with all the habitats that were identified. We used both model distribution with generalized additive models and then expert knowledge in the shallower areas where we didn't have environmental variables because the vessel could not access those areas. And in terms of the reefs, we have a 5.3% of the whole area, which is small compared to other sites of Spain, but we also have the bend affected area as well as caves. And then, as Christina pointed out yesterday, the pink area, which is what I characterized as 1110, which is the sandbanks, should be not taken into account because it's still a fragile topic, and it should be taken as proposed 1110 habitat. Thank you.
Thank you. Now I call Hussein Bazairi to talk about the current state of the marine biodiversity in the National Park of Al Hoceima.
Thank you very much. Well, the National Park of Al Hoceima is the unique marine protected area declared officially by Morocco in the Mediterranean. Real-level data on the marine part was produced in the framework of the project MED-MPA in 2002. And here in the framework, a new recent project, MED-K Habitat 2, with the support of SPARAC, we had the possibility to study in detail using a multi-approach with many scientists from Spain, from Morocco, to study the biodiversity in the marine part of this national park. So we used many approaches by sonar, yeah, to map the benthic communities between 0 and 50 meters. We had produced also bathymetric maps and so on. And it was also the opportunity to characterize some key habitats in the area, Coralligenous and Symposia nodosa meadows. Although to do the census of some endangered species, mostly Patella ferruginea, and to do some fish visual census in the same pod that were prospected in 2002. So overall, our intervention revealed that this area hosts about 49 protected species, in addition with the 27 habitat protected by Barcelona Convention using the new reference elaborated by SPARAC in 2019. And as a conclusion, it's clearly, we can clearly say that the national park deserves its status as protected area in SPAMI and at a regional scale. And this updated scientific data provided will help surely in the elaboration of the management plan that's in its final state. So here you have two figures and you have a QR code, so you can use it if you interested in to see summarizing the field work, how we work, and the video on the main biodiversity in the marine park of the National Park of Al Hoceima. Thank you very much.
Now I call Professor Giorgio Bavestrello to talk about the benthic paleo assemblages of the Seca coral. So from this side.
Okay, thank you very much. Okay. From 1875 and 1878, in front of the small village of Sciacca in the Sicily Channel, 3 large banks of fossil red coral was discovered. The amount of red coral was incredibly high. In 20 years, fishermen collect 18,000 tons of raw material. The way of formation of the Sciacca banks was for a long time a puzzle for marine biologists, but also the paleo community living in the Sciacca bank, including red coral, was until now practically unknown. Okay. The Sciacca coral was present as spread branches of red coral in the sediment, but also as conglomerate that help us to better understand the structural diversity of the community. And here we may see the main component of this conglomerate, obviously the red coral, the bivalves, Spondylus gussoni, brachiopod, Megerlia, some scleractinian like Caryophylla, but most important, Madrepora oculata. This finding was really interesting because we virtually do not know place in the Mediterranean Sea where red coral and Madrepora live together. The only place is the Cassidaini Canyon in the French coast. Here we have made an estimation of the age of some component, and as you may see, the age of red coral branch extend for more than 3,000 years before present, and the same for Caryophylla. But Madrepora is restricted to a small band, 1,000 years, that we may know as the Madrepora oculata age. So another possibility is to study the boring sponges living inside the coral by the shape and the size of the scars. Sorry, I know on the wall of boring chamber, and probably the species that bore the Sciacca coral is Siphonodiction coralli rubri. Next one, please. But we may ask if conglomerate with red coral are present only in the Sciacca. We think so, but recently we have found a big conglomerate from Sardinian water, and probably more interesting, the last please, in the collection of the pioneer of marine biology, Lazzaro Spallanzani, at the end of 18th century, collect this conglomerate with red coral in the Messina Strait. So the study of this material gives us a very good opportunity to study the paleo community with red coral in all displayed. Thank you.
Thank you.
Okay, thank you.
And now I call Martina Canessa to talk about the influence of substrate lithology on benthic assemblation in dim light condition.
Hello, good morning, everybody. And first of all, I'm sorry for the inconvenience that my presentation caused. Today, I show you a brief presentation about a study that follows previous studies about the distribution and the occurrence of species related on benthic assemblages in coralligenous reefs in the Tavolara MPA in Sardinia. And previous works highlighted the primary role of the substrate lithology in driving the structure and diversity of sessile zoobenthic assemblages in several ecological contexts and at different spatial scales, especially in the basal layer made by Coralligenous coralline algae. And this investigation was done on benthic assemblages settled in shadow habitats in dim-like condition characterized by the Leptosamia proboscifacies, settled on limestones versus granite sites. Our results confirm the previous ones, and firstly, the different abundance of Leptosamia proboscis, 3 times more higher on limestones, the presence almost exclusive of coralline algae and Peysonelia CPP that sometimes overlap the sclerites of the sunset corals, and the wide presence of branched calcified bryozoans on limestone that resulted dominant both in terms of diversity and numbers. In contrast, this is the situation presented on granite sites where Leptosamia is low abundant. Sponges, both encrusting and massive erected, dominate, and a low number of abundance of bryozoans, except for the chitinous and not the calcified Cartella papirea. So this is a confirmed results of previous work that we done, and it is a challenging topic for acidification and temperature increase that affect severely the basal layer of coralligenous reefs. Thank you so much for your attention.
Thank you, Martina. And now we have Celeste to talk about biological indicator of climate change in Andalusian coast.
Thank you, Atef. Hi, my name is Celeste Dominguez, a PhD student at the University of Seville. Here is a work related to my PhD topic on climate change. The objective of this work is to establish a baseline study of the mineralogy and calcium carbonate production of secondary calcifiers like serpulid balan, mollusks, or calcareous algae. This is to determine the response of these biological variables to sea temperature, as well as to CO2 system variable like alkalinity or CO2 partial pressure. This is important because an increase in the CO2 pressure affects the oceanic carbonate system. The study was undertaken in Andalusian coasts like Algeciras, as well as in the city of Ceuta in the north of Africa, both in the Strait of Gibraltar. In these two locations, at depth of 5 to 7 meters, 90 structures were installed for its location. These structures were made from PVC plates, as you can see. As you can see in the figure. Each structure had two plates facing up to create different environments. 1 year later, we took from the water, dry and treat in the laboratory with acid to eliminate the organic matter. Meanwhile, throughout the year, we take environmental data from sampling zones. and we analyzed the amount of carbonate and mineralogy of each structure. When analyzing the result, we observed that there were no significant difference between the two localities regarding the pH, but if we observe the calcification, the mineralogy and the temperature were significantly different. I would like to highlight that we are data from 3 years, but here we only analyze the first year. We expect to reinforce the significant difference when we look at the remaining years. So thank you for your attention and your time. If you have any question, you can ask me now or later. Thank you.
Thank you, Celestine. And now I call Gemma Donato to talk about biodiversity associated with the coralligenous in the case of columnar build-up at 36-meter depth.
Good morning. Good morning to everyone. I'm Gemma Donato from the University of Catania, and I'm going to talk about biodiversity associated with the Coralligenous, the case of a columnar buildup at 36-meter depth of Marzamemi. As we know, Coralligenous is a priority habitat for the Mediterranean Sea, but its nature and associated biodiversity are still not adequately known. Within the FREEZE project, Cresci Blue Reef that was showed yesterday by Professor Antonietta Rosso, we focused on coralligenous biodiversity associated with the canopy of a buildup, aiming to understand which taxonomic groups inhabit it and their species richness, specifically for bryozoans and serpulids. The studied buildup was collected in August 2021. All direct canopy forming organisms on the buildup surface were picked, identified, and counted. As a result, the surface of the buildup is densely covered by an algal bryozoan canopy. Algae are mostly represented by partially or fully calcified perennialiaceans and erect macroalgae. In the canopy, bryozoans are only represented by large, up to 6 or 7 centimeters high colonies of Margaretta cereoides. On the top right, we can see some examples of this algal bryozoan canopy. The picture A shows the Margaretta cereoides, on which bryozoan Margaretta cereoides, on which are settled serpulids. And picture B shows the algae Flabellia peciolata, on which are settled also serpulids. In fact, numerous species belonging to different groups settle on this canopy. We have some example like the bryozoan Celeborina caminata, the serpulid Spirobranchus triqueter, the foraminifer Miniacina miniacea, the mollusk Vermetus granulatus, and the ostracod Tenedocidere prava. As conclusion, the research points to a high species richness for bryozoans and serpulids, largely above the 9 and 7 species found on a previously studied columnar buildup from the same area. Though comparisons are difficult, bryozoans and foraminifers from this single buildup account for more than 1/3 and a half of the total species richness known for the Mediterranean Sea. So thank you for attention. You can follow us on Instagram if you want.
Thank you. Now I call Laura to talk about multi-species distribution models of Gorgonian in the coralligenous assemblages.
Thank you. Thank you. Hi, hello again. I'm Laura Picarola from the MetroCover Research Group and the University of Barcelona. And I'm going to talk about the multi-species distribution models of Gorgonians in coralligenous assemblages, which are a preliminary results. As you know, the species distribution models have been recognized as optimal tools to predict the species distribution. And our aim is to assess the probability to occur of 4 species of the Mediterranean Coralligenous assemblages that has been recognized as key habitat-forming species, which are the Corallion rubrum, the Unicella singularis and Eulicella cavolini, and Paramonifera clavata. We use different data sources, one from Core-MedNet that Cristina Linares presented you yesterday, and then other data sources from data gathered in OBIS, and then from different citizen science sources as Observadores del Mar or Reef Check. Using all this data, we have more than 70,000 occurrences from these 4 species. And then we use different types of predictors to predict the potential distribution of these species. The predictions were from IMONET, Copernicus Marine Services, and some of Biologia too. And we use the most refined resolution in that big regional geographic scale of the whole Mediterranean basin. So we calibrated the models and evaluate and select the most fitted one with each one of the species, and then used an ensemble model to obtain a projection of each one of these species. The main results show the probability to occur of these 4 species. Here we put the geographical extent to 150 meters depth in order to see the coastal probability to occur of these species. As we see, this is a preliminary results, and the 4 species showed similar habitat suitability distributions in these 4 species, and the most probability to occur were in the northwestern Mediterranean and Adriatic Sea, and also in Aegean Sea. But we have to say that we find some occurrences, true occurrences in the southern Mediterranean. So maybe if we had these occurrences, we had more probability to occur in that areas too. So this is one of the limitations to be considered. And then that this ensemble projection predict in some areas high probability to occur, and these areas we know that are not suitable for these species, but because maybe they're sand or mud areas. So we have to project a better predictions that it requires most accurate layers and expert criteria to find the most resolution predictions as we need. So thank you for listening. If you have any questions, we can talk later.
Okay, thank you, Laura. Now we have Ivan Guala to talk about distribution and study of Coralligenous assemblage in Sardinia.
Okay, thank you, Atef. Good morning, everyone. This poster reports the main results, the most current, most recent results on distribution, spatial distribution, and variability and ecological quality of coralligenous assemblages in Sardinia. We work on both coralligenous banks and coralligenous cliffs. And the activities were carried out along the northern and western coast of Sardinia. You can see the map in the background. Coralligenous banks were mapped by using acoustic tools such as echo sounder, multibeam, and side scan sonar, and using a ROV images as a ground truth. And images by ROV were also used to assess the spatial variability of these assemblages at multiple spatial scales, and also the ecological quality applying the Coralligenous Bioconstruction Quality Index. Coralligenous cliffs were sampled by scuba divers, on vertical substrates at 35-meter depth, and the perchance cover of main species were assessed to identify the spatial variability and also the quality of the assemblage by using the ecological status of Coralligenous assemblage index. Results: the map distribution of the Coralligenous is the red spot in the map. It covers more than 430 square kilometers of seabed along the continental shelf of northern and western Sardinia. It is about 9% of the continental shelf at the depth from 40 to 160 meters. As to the spatial variability, we found a small-scale variability for Coralligenous banks due to some local context-dependent taxa in some geographic area. As to Coralligenous cliffs, we found a large-scale variability, probably due to biogeographical partners rather than the distance among the study areas. As to the quality of Coralligenous, we find some inconsistency between the two indexes assessed in the same geographical areas. Of course, these two geomorpho types have different specificities, and this suggests us to consider both geomorpho type when we are planning monitoring and assessment of quality of Coralligenous. And so the main conclusion of this work is that we found some useful information for meeting the requirements of main European directive and Barcelona Convention. And it seems to me that the differences between the two geomorpho types are in line also with the discussion we had yesterday about the need to consider different aspects of Coralligenous when we are planning, monitoring, and study on Coralligenous assemblages. And so not easy to summarize in 3 minutes 5-6 years of studies, so I'm available for any question later on. Thank you very much.
Thank you, Ivan. Now I call Gusto to talk about contribution of Calcareous rotista to bioconcretions of the honeycomb.
Okay. Good morning. I am Giusto Lobue from the University of Pavia, and this is a brief overview of our work in on contribution of foramiferal tests to bioconstruction of Sabellaria alveolata. Sabellaria alveolata is a sedentary gregarious polychaete that builds different size and dimensions bioconstructions, which are created by arenaceous tubes packed together and built by agglutinating by sun-sized granules. Is known how among these granules, there are different kind of biogenic fragments and also foramiferal tests. For this, we tried to understand if the foramiferal tests were or are chosen as function of their morphology or simply of their abundance in the water column. With this purpose, we created an experimental design involving 2 sampling sites in the southern Sicily. At each site, samples has been collected from the sea floor and from the bioconstruction, and all the samples has been dried and prepared as washed residue to be quantitatively analyzed at the stereo microscope. Here, the data collected were about the foramiferal taxonomic identification and the morphogroups, in which they were all the foramifers were classified. 3 bioconstruction samples has been left intact to be epoxy embedded and prepared as polished section to be analyzed at scanning electron microscope. We found that in bioconstruction, in the bioconstruction samples, the quantity of the abundance of the foramiferal test were higher than in all the sediment samples in both sites, and inside the bioconstruction, the 2 morphogroups with correlated genera that were more abundant were the spherical and the biconvex one. In other words, we probably there is a link between the weight and morphology between the spherical and biconvex morphology of foramiferal tests that are changing their buoyancy performance, making them more available for the polychaete to be captured. And thanks for your attention. If you have any question, we can talk later.
Thank you, Giuseppe. Thank you. Now the floor is for Vesna to talk about Axinella in the MPA of Stari Olsini in Montenegro.
Hello to everyone. Well, I was involved together with my colleagues in the habitat mapping of the newly proclaimed marine protected area Stari Olsini. And from about 900 hectares of the area along the about 600 meters of the coast, we find something that is not typical Coralligenous, but in that area we found the real animal forest of the Axinella cannabina. As you know, it's protected spawn species by Barcelona Convention. It is protected in our national law, in some other countries. So we were exploring that area, and practically we found that the forest of Axinella is in a very narrow belt of the few meters wide and like 600 meters long belt of the coast, where the rocky bottom is finishing and sand is continuing. And we are very happy that practically this area is now in the second zone of protection. So it means there is no anchoring and there is no allowed fishing that is impacting the bottom. Because we found out that the density of those Axinella was almost 3 specimens per 1 square meter. And the average height was about 40 centimeters, but some specimens were over 1 meter high. We will see later what is going on with the mucilaginous, with litter, with different impacts like invasive species. But for me, it was particularly interesting that I couldn't find any other data to compare with these. And also this is, although this is the protected species, I couldn't find any data of the density of the population of this species. So if you have any information, please let me know. And for sure, I will check all the databases that we heard these days that are collecting information. Thank you.
Thank you, Vesna. And now the floor is for Victor to talk about cumulative effects of marine heat waves on Unicella in Mediterranean marine protected area.
Thanks, Atef. Hi, everyone. I'm Victor Arenas from the University of Murcia. I'm here to present the work titled Cumulative Effects of Marine Heat Waves on Unicella singularis in a Mediterranean marine protected area. Specifically, we here report 2 events of mass mortality affecting white gorgonian in the Cabo de Palos, Islas Hormigas MPA, in the southeast of the Iberian Peninsula, southwestern Mediterranean. Briefly, the white gorgonian is annually survived in this MPA since 2005, where around 300 colonies are annually assessed. When 2 variables are the proportion of necrotic tissue per colony and the proportion of affected colonies by necrosis. All this in parallel with the evaluation of the water column temperature. Temperature records are taken by in situ hobo sensors set up within the MPA at 4 different depths, 5, 15, 25, and 35 meters. And these data are used to characterize the thermal conditions and the detection and characterization of marine heat waves following the methodology developed by Hobday and collaborators. As a result of this long monitoring program, we were able to detect and describe 2 events of mass mortality. The first occurred in 2007, when damage extension per colony, which is around 1% in healthy population, reached 13%. And population incidence level, which was close to 60%, contrasting to the around 5% of reference period between 2005 and 2006. The second and stronger event took place in 2018, when damage extension was near 40% and incidence level was around 70%. In addition, although it was not quantitatively assessed, the red gorgonian, Paramuricea clavata, was also severely affected during both mortality events, as you can see in the photos. Regarding the evaluation of temperature analysis, 2007 had a really warm late summer and early autumn at 35 meters depth, when a marine heat wave of 11 days was detected from August the 24th to September the 3rd, when temperature eventually exceed 25.5 degrees. 2018, in contrast, was very dynamic. August mean temperature was the coldest detected in the study period, around 16.5 degrees, and caused a pronounced thermal shock when temperature increased beyond 26 degrees for 5 consecutive days, causing a marine heat wave between September the 12th and September the 19th. And temperature also remained above 25 degrees till late October.
To conclude, this study adds to the evidence pointing out the extreme temperature anomalies as potential triggers of mass mortality events. And it demonstrates the utility of acquiring and analyzing high-resolution biological environmental data so that observed mass mortality events and thermal anomalies can be linked. That's all, and thank you for your attention.
Thank you, Victor. I call Camelia to talk about mitigation strategy of the red coral to face climate warming.
Good morning. Sorry for the voice. So I'm Camilla Roveta from Polytechnic University of Marche, and I'm here to present possible strategies adopted by Corallium rubrum to mitigate the effect of climate change. So it's known that modular colonial organisms can respond to environmental factors using phenotypic plasticity. But when the environmental changes occur abruptly, the adaptive capabilities of organisms can be limited, especially in the aquatic systems, leading to frequent mass mortalities. Moreover, considering and following the temperature sites rule, a decrease in body sites was proposed as a common response of organisms to face climate warming. In this context, we wanted to present evidence in changes not only in the total number of colonies, but also in the colony sites of different populations of the red coral from various Italian locations, suggesting autotomy and resizing as a possible mitigation strategy against the rising temperatures. Different photographic surveys were conducted in 3 locations, Italian locations, Porto Ercole in the North Mediterranean, the Portofino MPA and the Gallinar Island in the Ligurian Sea. In Porto Ercole and Portofino MPA, photographic survey were conducted at a distance of several years, taking picture of the same cliff with the same perspectives, while in Gallinar Island, we focus on single transplanted colonies that were monitored yearly from 2018, the year of the transplant, to 2022. From all the monitored side, we estimated a few parameters, the total number of colonies, the colony sites, and also the number of apexes of each colony. From the picture taken in Portofino and Porto Ercole that were covering an area of about 20 meters square, we observed a significant reduction, especially in the colony sites and in the number of apexes of each colony. And while a slighter decrease was found also in the total number of colonies. From the Gallinar Island, during the 4 years of monitoring, the colonies were resulted healthy and vital, but as observed in the other location, they suffered a reduction in sites and also the loss of several branches, as shown in the figure. So these results suggest that maybe differently to previous mass mortalities, especially like the one of the 99 or 2003, for which we had almost an octocoral mortality rates of 100%. During the last thermal anomalies, the coral density may not suffer so much in terms of total number of colonies, but instead the colony sites can get strongly affected. Moreover, under laboratory condition, the changes in temperature and salinity have been found to induce fragmentation in red coral branches through autotomy. But it was seen just as a new type of asexual reproduction, ensuring the population survival after an environmental stress. However, even though we found this also in the field, especially in the marine protected area of Portofino, of this fragment detached from the colonies, as in the previous studies, we did not have any evidences of the resettlement of this portion, thus suggesting that the red coral can use autotomy and fragmentation to reduce their size and to mitigate the effect of climate change. Thank you for the attention.
Thank you, Camille. And now, Thanos to talk about first in situ assessment of red color population and the agency. We have to use this computer. This one.
Okay, hello everybody. It's great to be here, and I'm really happy to present our results on the presence of Corallium rubrum in the Aegean Sea on behalf of a group of great friends and collaborators. Now, what is the particular case of Corallium rubrum in the Aegean Sea? The red coral in the Aegean Sea has a notoriously deep distribution. It is bathymetrically distributed below 60 meters, so it is quite invisible to conventional scientific surveys. Now, officially, there is no harvesting since 1997, which means that there are also no landings data regarding its populations. There is no current population assessment, and the last, actually the only scientific study based on field data was conducted in 1989, which is 30 years back. So there is a huge gap in information, and this gap was acknowledged in the meetings of the ongoing GFCM research program on Mediterranean red coral, which also had the initiative to fund a preliminary survey in the Aegean Sea to assess the current status. Now, our field campaign was concentrated on 2 candidate sites with a bathymetry of 50 to 60 meters, which were the most accessible we could get and with the highest probability, according to all the information acquired so far, to actually find populations, living populations of red coral. So on these sites, we performed stereo video recording over 30 meters transect. And we used this video a posteriori to assess colony size and density from the footage. Now, moving on to the results, we found density, a mean density of 10 individuals per square meter in one side, but on the second side, this density was 10-fold higher, reaching an average value of more than 100 individuals per square meter. This was quite spectacular to us. This was a really dense population. And regarding the sizes, we could also see that this second side had some colonies with a greater basal diameter, extending more than 1 centimeter. So moving on to the conclusions, we can say that based on the results on these 2 sites, the coral density, height, and basal diameter in the Aegean Sea falls within the ranges reported from the northwestern basin of the Mediterranean for populations shallower than 60 meters. And this can be attributed more probably to the fact that this area has been closed to fishing since 1997. So probably the populations had some time to recover. But of course, one cannot rule out that at least one of these populations was not discovered by harvesters. And I would also like to say that we were quite excited to find the red coral and actually have the data to support that it is living, it is there. And we really want to move on to more elaborate surveys, both geographically and methodologically, to actually answer the questions that this survey has provided to us. Thank you very much.
Thank you, Thomas. Now I call Yanis to talk about exploring the timescales needed to recover the functioning of the Mediterranean Coralligenous assemblage through active restoration.
Hello again.
Okay, perfect. Hello again, everyone. I will be presenting one of the latest studies as well that we did, where we looked at the recovery of functions in a coralligenous assemblage through the active restoration of, in this case, Corallium rubrum. So this is usually quite done in terrestrial systems. When they restore, one of the key attributes they look at an ecosystem is if they are recovering functions, and it's something that it's not so much done in marine systems, but we think it's important that we start implementing. And this study started in 2011 when Catalan authorities catched almost 15 kilograms of illegally harvested red coral. They were given to the MedRecover research group, which then performed a transplanting experiment within the Medes Islands MPA. And they basically transplanted the colonies at around 15 meters depth in a rocky substratum. And the location was chosen due to there being a nearby native population of red coral. And throughout these 10 past years, both communities have been photographically sampled, from which we, through the PhotoQuad software, obtained the coverage of macroventhic sessile species. And then using trait-based approaches, we basically translated this taxonomic diversity data to a functional one by grouping together all species into functional entities, those that share the exact same coded value for 12 traits. The 12 species-specific traits selected are based on a previous study by Gomez-Grath et al. And are highly linked to the ecosystem services of coralligenous assemblages. These functional entities, then through multi-dimensional techniques, we can calculate a functional space. Here, very briefly, you can see how the different traits are distributed around this functional space. And well into the results, we can see a function and shift in the functional identity of the restored assemblage. This functional identity would be the mean value of the present traits. If we look at the winners and losers of these different functional entities, it's quite interesting seeing that there are some entities that have a bigger change than the red coral itself. And this would be an increase on encrusting sponge and hexacoralia, and then a reducing of the red encrusting macroalgae. And of course, the developing of the red coral colonies has also increased its presence. And from a functional perspective, then this means that we're moving into a slow growth, late reproduction, filtering mechanisms, colonial and heterotroph strategies, and tree-like massive and cap and corimbose morphologies, which we usually associate to those macroventhic habitat-forming species that have important ecosystem services, such as the habitat provision or the nutrient cycling. So, of course, the native site is not meant to be as the reference site. We just wanted to make sure that there was no other functional shift occurring within the MPA. But what we're seeing with the study is that the transplanting of red coral is speeding up the recovery of some key function for these systems. Thanks a lot.
Thank you. Now I call Kansu to talk about the sharp decrease of alcyonic assemblages following a severe mucilage event in the eastern Sea of Marmara.
Hello, everyone. My name is Cansu. I'm a master's student at Istanbul University. Today, I try to show you the effects of mucilage on soft corals and gorgonians in the Sea of Marmara. I bring my presentation in case.
Yeah.
Sorry. But it's not working. Sorry, do you have my link? It's probably down because it's not working. It's not working to pictures. Okay. I'm so sorry for the delay. Is it working now? Yeah, okay. So as my supervisor talked about before, Marmara is a very unique ecosystem. And in Marmara, we can observe Mediterranean mesophotic-like communities at shallower depths, especially around the Prince Islands, which we monitor there in order to understand the effects of mucilage. They're located near the Istanbul coast of the sea, and we survey 9 stations. Here I put some of the headings of that time. As you can see, mucilage covered the entire sea surface and it stays for a long time. It was really like a nightmare for us. But unfortunately, it didn't just disappear, but it sank through the sea bottom. And here I prepared this video to show you the mucilage aggregations on the water column and also the sea bottom. In Marmara, we nearly observe all typology of the mucilage aggregations like clouds, ribbons, flocks, cobweb, and blanket. They're huge. It's sometimes difficult to see each other during our dives. It's scary, and they stay for a long time. And as you can see, it nearly covered the whole benthic life. And as gorgonians, they have a fun shape. It's entangled with their branches and caused some serious damages. Here are the Eonicella colonies. And what we found, this area has been monitored since 2013. So we have the previous data. And as my supervisor talked, in 2015-2016 period, there's an excessive sedimentation. So their number has already been decreasing. But unfortunately, they try to regenerate themselves. But in 2019, mucilage hits. So their number decreased even more. For soft corals, they decrease after the sedimentation, 77%, and gorgonias decreased 19%. So if you look at the before and after pictures, I put 2 islands, Yassıada and Sivrada. Before we have like dense, healthy colonies, but now they are scarce. The distance between colonies is increasing, and they're not totally healthy, but necrotic and dead colony number increase. So Marmara is still resisting. For example, like the most common gorgonians for the Mediterranean endemic gorgonians, Eonicella cavolinis, Spinimyrcea claverini, Paramyrcea clavata, and Paramyrcea macrospina are the most common for gorgonians in the islands. Their number decreasing, but they still exist. But we need to take urgent and more serious conservation actions. So thank you for listening. This is our team. If you have more additional questions, you can find me outside. Thank you. And thank you. So sorry for the delay.
Thank you.
You can.
And now I call Hussein Bazairi to talk about the integration of Al-Husayma National Park into the CCIL Bioindicators Permanent Quadrant Monitoring Network of the Albaran Sea.
Thank you very much, Atif. Well, I will present you briefly this poster that normally will be had to be presented by Juan Sempere Valverde, but unfortunately he couldn't be with us. Well, this poster is about to inform you that in the same project, Medkey Habitat 2, in the National Park of Al Hoceima, with our colleagues from the University of Sevilla, we have initiated the program of survey program on Coralligenous in this area. So 3 points, fixed points, were installed in the marine part of the National Park of Al Hoceima using sorry. Okay. Using the Cecil bioindicators permanent quadrats approach, in this figure 1 and 2, we have you can find a code for a video that showed that give you elements about this approach that were used in this area. So overall, there's 3 points that were installed with also installation of data logger for temperature measurement, in situ temperature measurement in depths around 20 meters. So this first survey showed that there's 13 threatened species in the rocky shores prospected around this point, with 6 being present inside the quadrats, the permanent quadrats. And you will find in the poster preliminary analysis regarding the community, benthic community associated to this Coralligenous. And you see this from the eastern to the western parts of the national park. There's a clear differences in the community as a benthic conservation that associated the Coralligenous. So for any details, for any question, please don't hesitate to contact me during this session. Thank you very much.
Thank you. Thank you, Hossein. And now we have 2 online presentations. The first is from Isabella to talk about the first contribution on Corallium rubrum assessment in Sardinian water. Isabella?
Yes, thanks. I'm sharing my screen.
Thank you.
Okay.
Put it in presentation mode. Thank you.
Okay. Thanks. Good morning, everybody. I am Isabella Bitetto. I'm a PhD student of the University of Cagliari and a researcher of ISPRA in Bari, Italy. I'm going to present the first contribution on Corallium rubrum assessment in Sardinian waters. As already anticipated in other presentations, Coralligenous represents one of the most important biodiversity hotspots in the Mediterranean Sea. And it is also a key resource of Sardinian's water fishery. Unfortunately, it is currently showing a decrease in landing of the whole Mediterranean Sea, and it is an endangered species, according to IUCN. Despite the General Fishery Commission of Mediterranean Sea has put in place several safeguard actions for monitoring and protect this important resource, no stock assessment was carried out so far in Mediterranean Sea due to the scarcity of data on red coral fishery. Autonomous Sardinia region before 2012 launched observers monitoring programs that allowed the collection of the data on red coral in Sardinian waters. The objective of this study is exactly to provide the first evaluation of the status of this resource in northwestern Sardinian waters. We utilized several samples of Corallium rubrum colonies harvested by scuba divers, collected in the years 2012, 2013, 2014, and 2015 from 3 ports, Alghero, Bosa, and Fertilia, during this observers monitoring program. The basal diameter was measured for each colony, and from this information, growth parameters were estimated according to Marshall et al. 2004. Using these parameters, we derived the natural mortality using a combination of natural mortality methods. And finally, we carried out the assessment using length-based spawning potential ratio method. That is a method that is usually used to assess the stocks in data poor situation. So the age was estimated according to Marshall, and growth rate was estimated by area. So we found a different growth rate in Castelsardo respect to Alghero. The growth parameters were then derived utilizing a von Bertalanffy growth function with the nonlinear least square method. And then all this information was included in the stock assessment model. The results show that the fishery selects the colonies with the diameter that is above the size at first maturity. And this is a good news for the stock that has the time to recover and to replace the dead due to the fishery. Under the assumption of steady state, that is the main assumption of this type of stock assessment model, the stock spawning potential seems deteriorated because the spawning potential ratio is around 0.27, while the reference should be 0.5. So a moderate overexploitation situation is highlighted by our results. The fitting seems quite satisfactory. So the results, although they are quite old because based on the last year of information is in 2015, the results seems quite robust. To conclude, the decrease in red coral catches could be due to the prohibition of the ROV, the remotely operated vehicles, for the commercial catches, and also to the decrease of the number of licenses in respect to the previous years. The data collected under the observers monitoring program allowed to make this first evaluation in Mediterranean Sea about the stock status, highlighting that there is a possible moderate overexploitation for this resource. So it is the message is here that is very important to monitor this resource, and because it was very important to have this data available in order to have a first evaluation of the real status of the stock. So the message is that it is important to promote the monitoring program and the collection of this kind of data. Thanks for the attention.
Thank you, Isabella. And now we have another online presentation from Marco. Marco, you're online?
Yes. Hello, everyone.
Can you share your screen?
Yeah.
Thank you.
Can someone confirm that this works? Am I sharing the screen?
Yes.
Okay. Zoom in. Thank you all for listening to the talk today. My talk is named Slender and Stocky Morphotypes of Parazoanthus succinale species complex may represent two separate species. As the introduction section, there is a lot of evidence that show that slender and stocky morphotypes within this species complex are very different, not just in terms of their morphology, but also in terms of their life strategies and their genomic and metabolic profiles. And the question that we had in this study is whether slender or stocky morphotypes represent two separate species. And to answer this question, we collected 101 samples at 12 localities across the Italian coastline, which you can see here on this map. Populations of the slender morphotype are shown in yellow and of the orange in the stocky in orange color, and in total we had 68 slender and 33 stocky individuals. These samples were genome-wide genotypes for single nucleotide polymorphisms using 2B data, and we investigated the genetic differentiation between the morphotypes, but also the population structure within the two morphs. What we found is that at the between the morphotype level, the clustering is clearly really different. So all the slender and stocky individuals cluster separately, and we measured extremely high FSD values between them, ranging between 0.8 to 0.9 for each of the stocky slender population comparisons. And these values are much higher than what you would normally see for cnidarian species. They are shown here on the heat map, and these are the p-values, which are always significant. Within the morphotype level, we observed that the widely distributed slender morphotype showed a higher population of gene flow and population admixture. So all the populations are clustering here. The examples are those from Alassio and Portofino. So these here, which are often, this is often the area where we identify gene flow for other species as well. And then within the stocky morphotype, we found that they exhibit a higher level of genetic partitioning at the regional scale, and the populations are much different. To conclude, the study shows very clear differentiation between the two morphotypes with extremely high FST values, and these molecular observations do suggest that they may be two separate species, but this research should actually fuel for future multidisciplinary studies to examine whether we really do have two separate species. Yeah, this is just a graphical abstract to conclude. Thank you.
Thank you, Marco. Now we have the last presentation from Saïd Belbesha. Saïd, tu es là? Saïd. Je pense qu'il a toujours, I think he has always the problem. Saïd, est-ce que tu peux ouvrir ton micro, s'il te plaît? Ok, Saïd, on arrête pour le moment et on va essayer de résoudre ce problème et peut-être revenir tout à l'heure. Ok? Okay, so the poster session is finished. Just give us a few minutes to the evaluation committee to evaluate the oral and poster presentation and identify the winners. Thank you. Okay. Merci. Mais la Chine, non, je ne suis pas sûr. Même pas dans une drage. Please, for those who hang their poster for the Coralligenous, they have to take them out because we have the dark habitat symposia that will start in few minutes after the launch, and people want to hang their poster, please. Colleagues, finally we have the names. So the SPARAC would like to ask the scientific committee of the Coralligenous to come with us here in the podium, and I call Carlo Bianchi, Hossein Bazairi, Joachim Garabou, Mr. Leonardo Tonesi, and Mrs. Vesna Masic. So ladies first.
So Vesna, thank you for your tremendous efforts and for the quality of work during this, before and during the symposium.
Thank you.
So congratulations. This is a very simple and symbolic recognition that you can take home and remember these important events.
No, thank you for calling me. It's always the pleasure, and I think it's we will remember all these discussions and take-home messages. So we will be continuing our work. Thank you.
All together.
Mr. Hossein Bazairi.
Yeah, ladies first, and then by alphabetic order. Okay. So Hossein, thank you very much, and congratulations for everything, for the efforts before and during, and we will continue together. Thank you.
Just a word to say that I was very pleased to collaborate with our professors, our colleagues, and it was a pleasure. I hope that we have done nice work, and for sure we'll be here for another time. Thank you very much.
Thank you. Professor Bianchi.
No, Professor Carlo Bianchi.
Thank you very much, and congratulations.
Thank you. It has been a pleasure to collaborate with all of you, and I'm very proud if you say that our effort was beautiful. So thank you.
Thanks a lot.
Thank you, Professor. And Joachim. Kim.
What do you prefer, Joachim or Kim?
Kim for the French, so we are among French. So,
so thank you also, and thanks for coordinating this committee, this bright committee, even though Coralligenous are not easy to handle.
No, not easy to handle.
And we are not done with them.
No.
A lot of work before us. Mm-hmm. If you want to say.
Yeah, I want to thank the SPARAC for inviting me to be part of this community and making it a little bit bigger. And thank you for my mates, colleagues in the scientific committee. I know that I was not a really good coordinator of the scientific committee because it was a little bit messy. We have a lot of things to improve for the next time. We'll learn a lot of lessons here. So, but thank you very much for making it possible. Thank you very much.
Thank you.
Mr. Leonardo Tonesi.
Last but not least. Thank you, Leonardo. Thank you. I know that you have worked with a lot of patience. So congratulations and thanks very much.
Thank you. Thank you, Caraspa. Thank you, Director Calilatia, for the important organization and then also to invite me to be, and then in fact I'm proud to have collaborated to this important initiative for the Mediterranean for the conservation of Coralligenous. Thank you very much.
Thank you.
And now the name of the best oral communication goes to Sandra Ramirez Calero.
You want to give? Why not? Okay, so congratulations. I know you are very proud of what you have done. We noticed that, and we really appreciate your patience and your commitment. So keep going. You have a small surprise in here. If you want to say a few words.
What can I say? Thank you very much for this prize. This is very rewarding in terms of my early career, and I'm honored to be here. So thank you very much for this, and let's just keep working together for the conservation of our ecosystems. So thank you.
Thank you. And for the best poster presentation, the prize goes to Kansu.
Congratulations. Even though in the 3 minutes, actually, 3 minutes presentation, it was not very fair to present such great work, but we really appreciate what you have done. So congratulations and keep going.
Thank you so much for your consideration. I am very passionate about my field and I love Marmara. And I just want to thank my supervisor for her support. Thank you so much.
Thank you all. So I think now we can step forward and go to the conclusions of this symposium.
So Kim.
There has been a lot of things said during this one day, in 2 days. I'm not going to read all the recommendations because it's going to be long and I know that everybody wants to go for lunch, me too. So I'm just going to put some highlight the main topics about the recommendations that are in the list that you will find after, no, in the web page of the recommendations in the proceedings of the. So the first thing that we realized, or that we have been discussing, is that there is still, despite all the efforts, we need to put more efforts on acquiring basic knowledge on the biology, on the taxonomy, and the functioning of the Coralligenous and the other calcareous bioconcretions. So this is one of the things, and there are several recommendations on this. We have been seeing as well that there are a lot of impacts affecting these relevant and biodiverse habitats. And what we need is to reinforce all the instruments and tools that we have to promote their protection and good environmental status. Then there has been quite a lot of presentations on how to assess the good environmental status, and it's still an issue, and there are several initiatives and processes ongoing. And one of the recommendations that it seems that from the discussions that we have is that we have to work together to try to find this. And despite that, the IMAP implementation is not as good as it should be. It's a process that we need to encourage to follow up and to implement. The other point that has been also mentioned is about the collaboration and all these tools that we can put at the service of the collaboration initiative. There has been some examples of databases, or online databases, the T-MedNet, the CorMedNet, and others. Another point that is It's about the citizen science, that it can be a really useful approach for both, for acquiring this basic information, for raise awareness about the issues on marine conservation, and to enhance the dialogue between the different actors that are involved in the conservation of the Mediterranean Sea. And the last thing that we discussed, it was in the discussions as well, is about the need probably to have a communication campaign that probably the SPARAC can lead this, or with joining efforts on this, about the Coralligenous and other calcareous. And within this communication campaign, to make the broad society aware about these nice ecosystems and habitats, there is this issue about the valuation, how to speak with the different policymakers and things. So I recommend you to look at the recommendations and have on this. And thank you very much for all your discussions and inputs and presentations. I think that you are doing a great job. Thank you very much.
Thank you, Tim. So the closing remarks or speech.
Thank you very much, Kim, if you allow me, for these very precious concluding remarks that summarize more or less very briefly the spirit and what has been concluded from the presentations and discussions. I think that we have all learned a lot during these sessions. I personally am not a specialist in biodiversity, but I have learned a lot. And thank you for these take-home conclusions. I had the privilege to read the entire text of the conclusions, so I can make a very short and brief comments. It's sure that we have a lot of work before us, and the role and mission of SPARAC is crucial to coordinate at Mediterranean level, to support with the means and the potential that we have and we can mobilize. Our role is more to federate, to coordinate, and to put everybody working together, including citizen science. And we will have surely to work first on improving definitions and concepts in order to have very coherent terminology and definitions of the concepts, and also to have more data and more information and the overall distribution of the Coralligenous in the Mediterranean, in order to know how to prioritize the work and how to tackle it. So a lot of work before us, and I hope that this session, this symposium round will be a very useful one and starting point to improve our knowledge within this very important decade where we have all agreed on very ambitious targets in terms of biodiversity conservation, and particularly the marine biodiversity, the subject of our gathering. And I feel optimistic, but we have a lot of responsibility before us, including to mobilize and to have the right language to address these important issues. So thank you for your work, for the quality of the exchanges and the presentations, and for your presence and efforts. And I declare this symposium closed. Thank you for your attention.
So we break for the lunch and we should be back at 2 o'clock. 2:30, okay.